Should I?
Yeah, so I sent you the link via email with the itinerary, the Zoom link embedded into that itinerary.
I'm sharing my screen via the Zoom.
Yes.
Okay.
And then all you have to do is make sure your laptop is muted and your microphone is muted on your computer. Because I'm getting the audio from the microphone.
Okay. Got it.
You don't need to. Yeah. Okay. I'm right outside of the.
Testing 1, 2. Testing 1, 2, 1, 2.
Recording in progress.
I cannot share my screen right now. Basically, saying that host has disabled the screen sharing.
I need to log into my Zoom to allow something better.
Yeah, well, you're on the speaker. He's had a same issue.
From outside showed that they really wanted to Zoom as well.
Yeah, it's fine. No worries.
This was done like build out. A lot of other folks couldn't register.
Oh, no.
How do we access it? And we can't give them the Zoom option, but someone is always.
There's always the technology. Yeah.
So how are things? It's crazy.
It's good. It's busy, good, but
Good.
it's good.
Yeah, we really look forward to your talk.
Yeah, please. And I'm sorry I couldn't stay back tomorrow,
No, it's okay.
unfortunately. It's just that the project just started and they have all these I mean, because it's a 100-day challenge to design a complete vaccine. we have to give it the full attention because we don't know what they're going to select. Like, I don't even know what virus they're going to select. So,
So you try,
okay.
let's see, I wonder if she's got it
Yes,
set.
I can set it up. Thank you.
Yeah.
Yes.
Then, are you got any sound?
Yes, I do.
So on that other tab where it said share,
Mm,
Yeah, ability count.
Share.
Thanks, John.
where is it?
John, would you be able to?
You went past it real quick. There was a checkbox to share sound,
Stop share.
stop share,
Share screen.
share sound right there.
Ah, okay.
Yeah. And then you can hit share.
Probably I should also optimize it for video
Yeah.
clips. Can be quite interesting.
So, other than.
So you're gonna have to mute your computer.
My computer is completely muted. So
Yeah.
So where am I hitting that? Okay, so
I'm muted, but I think it's also because there's like,
continue with sharing. There we go. And so let's see, let's get to the sound part.
it's like a bunch of stuff that.
Marie, you're a co-host as well, so in case I'm not around. Marie.
Yeah, you got the solution as well, because it's not a title of the Zoom question.
I'm sorry.
It's not a title of the Zoom.
It's supposed to be sound in that.
Yeah.
Like,
Yeah, that'd be a good.
I think I'm gonna be the speaker and moving around the room.
I tried to make the speakers co-host beforehand, but UGA won't allow that until we actually get it.
Maybe I'm using your laptop.
My laptop is mute.
In previous versions, I demonstrated that GPT-4 could be directed to design parallelizable experiments.
I'm gonna make 5-6 results, okay? One, 4 for the speakers, 1 for the conference chair.
Okay, I think I'm good now. I think I'm good, right?
Yeah, if you just want to leave it, on the first slide, that'd be great.
Okay.
Yeah. That way we're ready to go.
But won't there be other things going on? Like,
I think the hope, welcome, not using anything. Go first speaker on.
yes.
Yeah, so. Might as well just leave it ready to go. There we go.
All right.
Let me show you these microphones.
I'm sorry, I didn't understand that you talk.
Yeah. From my understanding of the one. Let's just think about that. Basically, they come on.
Yeah.
When they go green, so about 2-3 seconds before they become live. So, anybody who in the audience who has a question needs to have a microphone for it to go out.
Oh, yeah, that makes
Yeah.
sense.
Yeah, so maybe they'll make an announcement, ask them to make an announcement at the beginning, to raise their hand if they have a question. So that way you can see who wants questions, so once they ask a question, the instructor answers the question, and we gets the next person.
So I would also moderate people who want to ask questions in Zoom. Should then mute yourself and ask the question for someone else to ask the question. In that case, I'm gonna keep the other microphone to the person designated to.
If you want to do that and you monitor the Zoom with the chat, you can tell the people online to chat with you their question, and then you just ask their question.
I think I'm gonna assign that to some part of the person.
Okay, we'll have them again, we'll have a microphone to the back, so yeah, all they have to do is turn on the mic, ask the question. Okay, yeah, that makes it a lot easier. But if that person wants to sit back here, that'd be fine.
If that person wants to sit back there.
Yeah, so all they're doing is monitoring the chat. Yeah. Yeah. Taking out. Very good. Okay, thank you so much. All right, bye. Hey, Elise. Yeah, whatever.
Right. It doesn't make sense. Yeah.
Hey, how you doing? That's gross. Yeah. I just wanted to.
And then the
You can post it. Because I don't think, general, student function.
I'm not sure.
It's the same Zoom thing.
Yeah.
And all.
Yeah, it's a whole Zoom for every all day. All right. Got it.
Good.
Great.
And the hardest one is gonna get out and get in those 15 minutes presentation.
Yeah.
And if you're in a room and you open up the Zoom link, make sure your laptop. So don't get it in.
You are right.
Yeah. So basically, all you can do is, when you're sitting down, if you want to sign in, you're not looking at the projector, basically doing the video out. Yeah. It says in the account, they're recording it onto their cloud. So, yeah. So I'll say to you, I got that. I think that is one almost. Do I take the microphone sound later? If you want one down first. Yeah, I got it. One, second number one, computer number two back here.
Hello. Microphone testing.
Microphone testing, 1, 2, 3.
All right.
I got that. I told you that I got that. Right there.
Okay, good, great. Got it, great, good enough.
I can say for souvenirs. Thank you.
Check. Hello? Is working? Okay.
So they're not gonna use that, right?
No, I don't think so. I think this is good enough, right? Yeah. Okay.
All right. Uh, Good morning, everyone. If I can have your attention, please take a seat. We're getting ready to kickstart the symposium. So welcome, everyone, to the IOB Special Symposium on AI and Deep Learning Applications in Biology and Bioinformatics. Okay, it's a little starter. Is that okay? Is that is someone in the backside? All right. Can we? Okay. All right. Good. So we'll make sure that doesn't happen. So I'm Kannan. I'm a professor of IOB and biochemistry here at UGA. So on behalf of my co-organizer Kaizhong Yi, the IOB faculty, staff, and students, I really extend you all a very warm welcome to this. What is a very exciting symposium that we've planned for you. So I also want to use this opportunity to really extend a warm welcome to our speakers who've really come from a long way to share their exciting work with us today, and also a special warm welcome to our sponsors, industry sponsors, 10x Genomics and Admira Health, for supporting us with this symposium. So I think this is really an exciting time in bioinformatics, where advances in computing are really revolutionizing the way we analyze biological data sets. And today you're going to really get to experience that with the talks. But I think when we conceived the symposium, I think that the goal was to really get people from very different disciplines to come together and sort of exchange ideas and explore new collaborations. So it's with that idea that we conceived this symposium. And I'm really glad that today in this room, we have representation from at least 15 different units. And just to name a few, the bioinformaticians here are working or sitting alongside computer scientists, biochemists, geneticists, engineering school, folks from engineering. We have students from pharmacology, wet med, ecology, public health, and infectious disease. So that's really a wide representation. And I think this will be an exciting meeting, and I really encourage you all to explore new collaborations. So without further ado, I really want to now extend a really warm welcome to our special guest, Vice President of Research, Dr. Karen Berg, who's really taken the time out of her busy schedule to be with us here today. She is a really distinguished scientist herself, and I don't think people at UGA need any introduction to her, but for our visitors, she is the Harbor Lights Chair and Professor of Small Animal Medicine and Surgery. She's done some pioneering work in cancer diagnostics and developing new technologies. And she's won numerous awards for her accomplishments. including elected fellow of the National Academy of Inventors, fellow and ambassador of the American Association for Advancement in Science. And she was recently named by President Biden as one of the 15 recipients of Excellence in Science, Mathematics, and Engineering Mentoring. So I'm really delighted and honored that she's taken the time to come and give us the opening remarks. So thank you, VP.
Great, thanks so much, Kanan. And please know there was no choice. Kanan has been very pesky in his organizational skills and has been on my calendar for months for this event. And I am indeed excited to be here to welcome you. So for those of you from the University of Georgia, welcome to Masters Hall. And for those of you not from the University of Georgia, welcome to the University of Georgia and our Athens enterprise. So we are delighted to have all of you here. I am noting there's a large gap between our wonderful invited speakers and other audience members. So don't be shy as we approach the breaks or in between to filter down and join one another in conversation. So AI, it is absolutely something that has been part of every conversation, whether you're at the gas station filling up or charging your electric car, or whether you're in the grocery store, or whether you are actually in the field of research. It is a household phrase, which is really exciting for us as we start to delve into where this might go. It is indeed what I consider bioinformatics of the future. And so we get to define the future, which is pretty exciting. The development and implementation is what I would also call one of the defining scientific questions of our time. Again, it permeates our society. Where is this going? What is this? How can we best handle this to our advantage? Obviously, we know folks are thinking in the opposite direction. You know, how can we use it for ulterior motives? And that's also our challenge, to figure out how we can use this for positive. It's absolutely critical because it could conceivably be applied for better or worse in almost any scientific, or me as an engineer, any engineering enterprise or undertaking that you can think of. And that's pretty staggering to think of being involved in a field that has that much effect and will really be the backbone of a lot of what we do in many other fields. And so our keynotes today, you'll hear some familiar concepts. So protein folding and spatial genomics. We know AI also very well from drug discovery, biological sequence matching, phylogeny. As we start to spin further away from those topics, let's think about biomedical engineering. That's my own field, and certainly we had a whole an international meeting last week where AI was a big topic, and it was the Biomedical Engineering Society. And so again, biomedical engineering, how about climate modeling or astrophysics and astronomy, or something that we are building here at UGA and is being built at other places, integrative precision ag. So agriculture technology that really spans across the disciplines as well. So it really is fundamental to so many different areas. So, and I don't think you have to be a specialist in any of those fields to appreciate that point. We know that AI will transform those fields through faster, more efficient data processing and analysis, and also providing really pathways as to where we might go in those fields. At the University of Georgia, just to give those of you who are visiting a little bit of background about our focus on AI, it's one of the very compelling reasons that we just developed a School of Computing. So that was, this is an initiative that we launched in the last year. This is a joint effort between our Franklin College of Arts and Sciences and our College of Engineering. So purposely very much an interdisciplinary activity. We know that in the 21st century, computing is firmly established as an arm of modern science. If one thinks about three-legged stools, which we do a lot in academia, we always talk about teaching, research, and service, but if you think about fields of study, you would definitely think of computing as being a solid leg on a three-legged stool, where we also have experimentation and we have theory. And AI certainly comprises both. So we're envisioning with the School of Computing a future in which our scholars will be collaborating with colleagues across the university and across the globe, around the globe. And that's, again, purposeful by design, that is a collaborative between two powerhouse colleges. We also have started a hiring initiative. So those of you at UGA are very familiar with this, but if you're not from the University of Georgia, I'll give you a little bit of background. In 2019, we launched a targeted hiring initiative for faculty in AI and data science. And then we expanded that last year based on our view into the future, that this is something that we really need to, we really need to wrap our arms around, and we need to do this in the way that we do best at UGA, which is across the breadth of the many disciplines that we have. So our goal is we will be welcoming by the end of the hiring initiative, 70 new faculty members in this area. They will be seated not just in the School of Computing, but across the campus in many other disciplines and departments. And so that, again, is really exciting for all of us who are working in the area of research and innovation. So hopefully the goal is they will be working with many of you. I know some of our new hires are here and also and or working with you all already. But the goal is that whether you're a UGA faculty member or student or staff member, or you're visiting from another institution, that you will be working with folks from that hiring initiative. So I would say we truly are living in a dizzying time because it's changing so rapidly, and you're at the forefront of that. So that's a pretty awesome responsibility, but also an awesome opportunity. There are global implications of AI, not just local or regional, but this is global. There are so many things that are developing globally that we think about when we think about the potential of AI and the accomplishments of that, whether it's our vaccines to combat, for example, COVID-19, or whether it's the improvement of patient diagnoses through the use of intelligent systems, or whether it's the extension of medical care into remote areas of our country or world. And you don't have to travel out of our state to appreciate the significance of that comment. But certainly around the globe, that's also a compelling opportunity. So it's really so large that it's almost even hard to describe the massive reach and depth that AI has and will have. So I know you have a lot to think about and a lot to talk about. I'm excited. You have 2 days, both hands-on as well as some listening today and some interaction, questioning, discussing today. And so I appreciate the fact that you have lots of challenges to talk about. And I appreciate the fact that you've all enthusiastically come together at 8 o'clock in the morning on a Monday to do that. So again, AI is something that it's not one of their many things that sort of the terminology comes and goes. This is not one of them. This is here to stay and is solidly a part of what we think about as we think about a research and innovation enterprise. It's up to all of you to actualize this, to really envision and think about where we might go and where we've been. And personally, I speak on behalf of our Office of Research and all of our innovation folks that we are here to collaborate with you to make sure that you are able to reach that vision. So you'll be showing us how it can be put to work for both the health and benefit of all of us as we think about folks on the globe. So a very big welcome to all of you. I hope you have an engaging and thought-provoking 2 days, and I'm looking forward to hearing the outcomes of that. Both as a fellow colleague, a researcher, but also as a member of society, as you unravel some of the possibilities and determine what the paths are forward. So again, a very big thank you for being here, and a very big welcome to all of you, whether it's to Masters Hall or whether it's to the University of Georgia at Athens. So with that, a big thanks, Kanan.
And I think we're getting ready to now get started on the science, and it's my real pleasure to introduce our first speaker, Arvind Ramanathan. He's a computational biologist in the data science and learning division at the Argonne National Laboratory. He's also a senior scientist at the University of Chicago Consortium for Advanced Science and Engineering. His complete biography is all printed, so you should all have as a handout. But I think he's doing some really exciting work, and I think his work at the intersection of data science, high performance computing, and biological data analysis is something you're more likely to witness in his talk, which is titled Integrating Generative AI with Automation and Simulation for Biological Systems. So let's welcome Arvind.
All right. Hope you can hear me well. Okay, good. So I'm going to be giving you an overview of things that are going on at my lab, my group, and a whole bunch of other collaborators as well as I continue with the talk. So this is about thinking where biology will be in the next few years. So there are some things that are like put together in terms of future outlook as well, but also things that we are currently doing inside the lab to kind of support that sort of future. So much of this project actually started with this idea that, if you take a look at this beautiful picture by David Kutzell on the cell, you can think of all these parts that are sitting inside a cell as parts, real parts. Like you can reuse them, you can rebuild them, you can customize them, you can do all sorts of things. And we are already doing that in some form. Like if you're going to design a vaccine, you have these neutralizing antibodies that you need to build. You have various scaffolding proteins that we are building, cellular compartments as such for drug delivery and things like that. All these different types of things that we are doing from bottom up or top down approaches are really impacting what, how we think of them as reusable car parts. And part of the story in some sense is impacting a lot of these varied applications, including antibody design or industrial production of metabolites, including beer hops and things like that. Then there is a lot of work in biomaterials and biosecurity, where people have been thinking of how we should reuse and reorganize these parts for better use across various applications. Within the Department of Energy, one of the things that they have been really interested in is this integrative platform, as they call for engineering these biological parts. So part of the process of this was to construct what we call now as a closed loop automated systems for engineering these biological modules. And these modules can be anything from transcription factors to biosynthetic enzymes to various chemical pathways to genetic parts and things like that. And we needed to build ways in which we could get high throughput tests of these systems built and tested inside the lab. And one of the unfortunate side effects of doing these types of biological questions is the fact that reproducibility is very hard in these things. And one of the fundamental ways we have been thinking about it is to approach it from a computer science perspective. So in computer science, if you think of the ways we have been building these supercomputers or various ways in which we have been engineering systems, one of the good things that comes out of it is that there is a set of inbuilt tools that actually talk about reproducibility. When I say two numbers multiplied, 2 into 2 has to result in 4, there is a way to validate that 2 into 2 is actually 4 on the computer chip that you're building. This is unfortunately not the case with biology, where you can actually have like probabilistic responses, and you need to be worried about that, which is good and bad in a certain way. So we wanted to see how we can make reproducibility as one of the first citizens of how we were designing our biological systems. A big portion of the study has actually been towards building systems that are more programmatic. That means that you can actually type code and you would actually be able to execute that biological experiment end to end, right? So you're going to hear a little bit about what Andrew has been doing in chemistry, and this is the equivalent of doing that in biology, where we have been thinking of how to run that from the concept of saying, if I provide the top level goal of saying, this is what I want, how do I ask my AI to actually like implement and execute it? And all of this is actually driven by predictive analysis toolkit that we have been building, and some of which has foundation models, which is the foundation for like building generative use cases. And then you have all these engineering systems that need to come together to make the robotics work on one side, write protocols on the other side, and then have them execute them on the final end of it. So why this is actually required. If you think of the current paradigm of how we have been working in terms of designing these cellular parts, there's a lot of work that needs to go in. So you start out with this massive workflow of taking all these parent sequences that you know. So you have to do alignments of them. You have to get all these scaffolds and active site identification, which in itself is a huge issue if you don't have well annotated data. If you're working with a new enzyme or new system, then there is all hell to play for that as well. Then you have all these combinatorial libraries that you need to build. That in itself is a self loop of like building them many, many times and trying to see if they work or not. And then you have to score these substitutions against your libraries that you might already have. And then you get these ensemble of ranked variants that you think might work in the world, and then say, okay, and let me put them through the high throughput protein screens or assays. Then you eventually select these sequences and then say, okay, I'm going to update my new parent sequence libraries. But if you just think about the combinatorics of the problem, this is actually kind of complicated, right? So only a very negligible fraction of the available sequence space can be really searched. This is supposedly 1 by 10 to the power of 117, right? It's an extremely small number to like really be thinking about it. Maybe in the logarithmic sense, it's a very big number, but you know, in this world, it's actually kind of very small. And if you were to think about the combinatorial optimizations that are involved, if you take an entire biological pathway, for example, this becomes even more complex. So the moment you think of actually exploring certain fitness landscapes, as they call in biology, of saying what is actually a good fit for this particular problem, we are not exploring all of the possible design space that is available to us. And if you're not using that, you're really in a very poor space of like fitness, and you're not really exploring what designs might actually be possible and what sort of new things that we can design. So as a consequence, this current paradigm of how we are running these experiments and designing new parts is really not scalable and also not sustainable. Just various problems with this. So what do we do to address that? And this talk is mostly to build those sorts of intuitions of what works and what might not work. So I'm going to talk a little bit about learning representations for complex biological data sets. This is mostly built on the fact that we want to do foundation models for genomes. I'm sure you're going to hear a lot about protein language models, which is all the rage right now. And there are several groups that are building. There is an open challenge going on. We are actually starting at the genetic level, which kind of makes it easier and also harder at the same time. I'm going to talk about the aspects of challenges that we are facing there. And now, once you build these foundation models for genetics, we have the ability to scale them for entire genomes. And I'm going to give you an example where we said, okay, is it possible for us to predict the next set of variants from SARS-CoV-2 as an example? And then I'm going to show you how the generative modeling capabilities can be used to engineer new proteins like malate dehydrogenase with certain levels of catalytic activity. The last half of my talk is going to emphasize on building what we call as embodied agents as scientific assistants for biological discovery in the lab. This is mostly based on work that's ongoing in the lab for building what we call self-driving labs, which means that there are no people inside it. We don't want humans that are involved in like basically running mundane experiments, but elevating their creativity to a certain level so that they can actually run better experiments. And also in terms of teaching robots how to write biological protocols. And then I'll talk a little bit about that application to antimicrobial discovery, and then conclude with my perspectives on what needs to happen. So first, in terms of biological information and hierarchy, I think this is something I'm just going to repeat it for everybody's benefit in the sense that, when you think of a single gene all the way to humans, there is a very neat hierarchy of how genes are organized. And within the gene itself, you have various portions that actually constitutes a functional part of a gene, which includes the promoter, you have the 5' untranslated region, then you have the open reading frame followed by the 3' UTR and so on. At the level of a single gene, any model that takes into account any sort of functional details has to pay attention to the fact that there is this intrinsic hierarchy of how that information is represented. So in this case, maybe the codon level tokenization or approaches like that will work well. That was our hypothesis when we started. So tokenization basically means that you're actually splitting up your input. So when you have a set of sentences, you have words that form those sentences, and those words are actually the tokens. You can also split it according to characters if you're more ambitious, but let's assume that we are simply keeping it at the level of the words. And this is what works practically in terms of the genomes too, where you can use these codon level tokenizations. You also have this ability to now then think of combinations of certain set of genes, which now becomes a pathway, for example, right? So these are collections of genes or contigs and ORFs, or open reading frames, at which level maybe certain things don't work very well because of the length restrictions on what you can tokenize. But you have the ability to basically sit and understand and say, okay, I'm going to collect them as a group of these ORFs, and then say, I'm still going to use the codon level tokenization, but working at a much larger token window. And then you can take it to an entire genome, which means that you need to work with full collections of all these genes or ORFs, which can then make it very difficult for existing models to work with. So there are new approaches to deal with it. I'm not going to touch the latter half of the story where we are dealing with entire genomes. I'll show an example and then leave it there for open thoughts. But this idea that hierarchical representation can really work is the foundation for what we call as genome scale models. And these models actually go beyond traditional K-mer models, right? At each level of the hierarchy, we tend to maintain information that is learned at the lower level, from genes to collections of genes to full genomes. But this allows us to scale with each level and tie the information back every time you're actually building the hierarchy. So this sort of a model, you can think of simply working with something like Markov random field or a Markov process. I'm sure you have used these hidden Markov profiles and things like that to do comparisons. But this is a generalization of the same approach with deep learning methods. Okay. So the genome scale models as such are fairly simple. You take an input sequence, break it down into a set of these tokens. These tokens are codons at the level of an individual gene. And then you have transformer layers that are put together, and you do a semantic embedding of that input layer, right? And what you get at the end of it is this semantic embedding, which is just a vector, a high dimensional vector. I shouldn't say it's just a vector, it's a high dimensional vector that basically represents some biological information at scale. And we'll see what that information is in a little bit. The advantage of doing it at the gene level or the nucleotide level is that we can use these codon tables to back translate whatever protein code it stood for. So it's essentially trying to mimic a protein language model. I mean, it has correspondence with the protein language model, but it has this information at the level of the individual nucleotides. So now these foundation models that we call as GenSlims or genome scale language models were built with a maximum sequence length of about 2048. You need to pay attention to that length because that's one that determines the effectiveness of the models at certain scales. Okay. And each of them were trained with all the way from 25 million to 25 billion parameters. We have now recently updated them to become more chinchilla-like scaling. If you have not heard of chinchilla, don't worry about it. You'll probably hear a lot more in the later talks. But chinchilla is actually a language model that was based on text. And they talk about scaling laws for this. We have actually built the same set of laws. And so for genomic data. And this data set constitutes about 110 million genes that were obtained from what is called as the Bacterial and Viral Resource Center, or BVBRC. So this represents one of the largest foundation models trained on open genomic data, right? Most protein language models are usually trained on the set of UniRef 90, which is probably about 45 million proteins or so, on 20 alphabets. This one is trained on our open data set of 110 million genes obtained from this particular data set that I talked about. And then you can now specialize these models at the level of an entire genome, which is what we call as the GenSlim or GenSlim without the foundation. And this is now 10240. The 10240 is actually a special number because it also constitutes the length of the COVID genome. So there are 30,000 characters in the COVID genome. So it happens to be in that sweet spot where we could say, okay, we can actually do it at the entire genome scale. But once you do that, you can only train on all the open reading frames at a time. It doesn't know how to put the context, that is how to put together the gene orders together. So we need some sort of a model that captures that hierarchy, which is part of this model that we call as the diffusion model that comes on top of this. So the diffusion models were also trained on the GenSlim models that I just talked about, but instead they also carry a diffusion parameter that allows us to basically reorder the sequences in the terms of seeing the genes. So that's actually another 2.5 billion parameters. That was just trained on the 1.5 million SARS-CoV-2 sequences that were seen just the first year of the data. So we're not presenting any other amount of data for it to train. And then we're asking this question, is it possible for us to predict the future strains or not? That's later, right? If you just look at how GenSlims actually work, they achieve what are called as the state-of-the-art perplexity, even with very short timing, short training cycles. So perplexity is just a measure for the number of guesses required by the LLM to predict the token of interest. So the next token of interest is what we are predicting. And a perplexity of 1 implies that it's a perfect model. And in general, if you ask me, my personal opinion is that perplexity is probably not the best metric for genomes, right? I don't want to like get into that discussion, but we had to show this because a lot of them ask us, what is the perplexity of the model? So we do show them. So yes, these models actually have good perplexity, but I want you to observe one thing, which is that the 25 billion parameter model is actually poorer than the rest of the other 3 models that we train. And that's mostly because we feel that it does not reach sufficient training. It's probably seen only like 11% of the overall data, which is kind of not enough for it to learn enough. So there's more work to be done there. I'm not going to show you results of scaling these models on our Cerebrus CS2 systems, which is one of the largest AI chips that's available out there. It's the size of a salad plate. If you have not seen these chips, it's a really big chips for training AI models. We actually use 16 of them in parallel to actually train the model. And we can actually obtain normal perplexities like what you're seeing here, but I'm not going to talk about that. It's just too much detail into the scaling. And most of the results I'm going to present today are based on the 2.5 billion parameter model, which is just the one that we thought was more stable from the time that we actually trained them. So these models actually reveal quite a few novel biological insights. So if you now consider, this is on the testing data, which is about 10 million sequences that was held out. This is not seen from the model's perspective. And each of the dots here is the latent space representation from this very high dimensional vector Z I was talking about in 2D. So you have to understand that there is a lot of play in this case when we are presenting the data, especially in these 2D representations. But what I wanted to show was that if you paint them with certain characteristics about the genes, for example, the sequence length, you can clearly see the separation between the low and the high sequence length sequences. So if I were to build a predictor from these models to say, can you get a predictor for the gene length, it will do very well. It's also doing something very well in terms of GC content, which is actually a hard thing to do if you think through the process of, why it is needed. So the GC content is a measure of adaptability of the entire genome to a particular environment. So if you're able to predict the GC content, then you're able to say something more about the nature of where these genes might adapt themselves. So maybe some are hypothermophilic, some are less hypothermophilic, and so on. It can also pick up on things like biochemical function. For example, if I say where are malate dehydrogenases located, it shows them in these 2 independent clusters. And these clusters are kind of interesting because one of them happens to have 1 isoform and the other one happens to have a different isoform. And again, we did not give this information to the model, right? We have not given any fold information. We have not given it anything other than the nucleotide sequence. The fact that it can sort of pick up these patterns from the data is sort of indicative of the fact that it is learning something about genome scale organization that we might not have really understood from the beginning if you just use traditional approaches. Right. So once you do that, you can also start asking questions related to the function level organization. Now I can say, where are my stress response and defense, defense and virulence proteins distributed, genes distributed? You can actually see that they form a nice cluster But many proteins also have multiple domains that have different functions associated with the same thing. So if you think about that aspect, there's like a lot of clustering here. But if you look at whether we are able to predict accurately the function, this achieves about 87% accuracy in terms of function prediction from just the language model itself, right? Now, the neat thing about it is that it can also distinguish between coding and non-coding sequences. Again, this is tested on eukaryotic genomic data and not on the prokaryotic genomes like what we were seeing from bacterial genes, which was part of its training corpus. So it can actually tell you what are coding sequences. CDS represents coding sequences. It can tell you the difference between tRNA, tRNA, mRNA, and rRNA. And what is even more interesting is that the 25 million parameter model that we were using, which is on the right-handmost corner, seems to be at least better than the hyena DNA, which was actually used to train much longer sequence contexts. So we are still in the process of testing all of this in the, in our data sets that we have, because I am not a eukaryotic geneticist. I don't know much of the data sets that exist out there. So there's a lot of learning going on in terms of like understanding what it means to do this. But the fact of the matter is that we are able to get at least like 98% accuracy with even the smallest models in terms of like looking at completely out of distribution data and then trying to understand what happens. I need to talk about the infrastructure piece a little bit here because, we say that these models are foundation models. Is it going to be available? So my student, Kyle, who's here, he basically planted himself in front of these 3 supercomputers. And the last one that you see at the bottom is Selene, which is NVIDIA supercluster of about 4096 GPUs. It's one of the largest supercomputers built for training these sorts of foundation models. And we're kind of happy because this was also the first time they opened it up for external use other than NVIDIA folks. So we trained them on their systems because we couldn't get enough computing time on either Polaris or Perlmutter, which is both supercomputers from the DOE systems. But once you get those foundation models, they actually use very, standard transformers, lightning and deep speed infrastructure, which is usually the thing that you'll use. And the code is very usable. So we actually provide you the entire training code base, the data sets and everything. You can download the data. Just don't blame me if it is 512 gigabytes for the compressed data, but it is available. You can download it. You're welcome to play with it. You can take the data, input any sequence, and you can actually like do a lot of things like what you do with your Hugging Face models. If you have not used Hugging Face, if you have not used transformers, just, sit and play with it for at least an hour. You will learn a lot. And it's fairly simple to use these Hugging Face interfaces to like create your own models and play with them. Right. Now let's talk about the fun things that we can do with GenSlims, right? So if I were to take a very naive approach and say, let's generate new gene sequences, this is based on just a beta sampler that's written on top of this embedding distributions that we get from our data and say, okay, now start picking up these sequences and then see if you're able to like generate new protein sequences. I'm going to show this on malate dehydrogenase because that's our favorite protein to work with. It's also a protein that we can do assays in like a few minutes instead of waiting for days. So that's the reason why we do it. But if you look at these sequences and then start asking the naive generation, does it fit anything that we understand about the evolutionary details? So if you were to construct this phylogeny, which is actually consisting of like 36,000 malate dehydrogenase sequences, you can see that these sequences that we start looking at black dots, these are here. I'm sorry, you can hardly see those in the picture. But if you were to generate those sequences, they start filling in those gaps where the sequences are not sampled from the real phylogeny. Does it mean that these are natural proteins? Does it mean that they are not natural proteins? We don't know that question. But what we do know is if you take them and actually test them out in the lab, that these generated models, they actually work very well. So from this set of generation, which was naive generation, we took about 96 of these proteins and said, okay, can we generate the gene? I mean, we take the generated DNA sequences, order the genes from the twist gene or any one of your DNA providers, and then test them in the lab. About 49 of them end up becoming good malate dehydrogenases with good catalytic activity. And of the 96, we were only able to test 49 because that was the amount of money that we had. But we can still go back and test the rest of them if, people actually say. And these proteins do share similarity as key sites that we predicted from the two, isoforms that I showed you, two dominant isoforms that I showed you. This is also based on open folding of these proteins and then trying to understand if they share those key sites. What I want you to observe here is that there are certain locations that are in the active site, but there are also certain locations that are much farther away from the active site that seem to be conserved, which is kind of crucial for the enzyme fold to actually function, right? So why not take this idea and try to do this as a ChatGPT app? Right? So a ChatGPT app is essentially taking human feedback and then asking your reinforcement learner to learn from those preferences and start generating new text. This is how most ChatGPT applications, actually all of ChatGPT works with that principle, that they use reinforcement learning and human feedback to guide the generation of new text. But we don't have human generation here, right? I'm not going to ask for new proteins by asking it to say, okay, is this substitution good? You can probably do that, but that's not very meaningful. So instead, we actually said, is it possible for us to use something like, experimental data as a way to give in feedback to the model? And I was not at all confident that this method would actually work. Like 100% guarantee. I said, no, this is not going to work. But we said, okay, let's take the genome scale models that we have built. We'll provide it with experimental data on whatever we observe in the lab in terms of activity, and then use that as reward functions to basically say, generate the new sets based on what we see. So one of the first experiments that we said was, is it able to generate a new sequence from scratch, which looks like malate dehydrogenase, but not exactly malate dehydrogenase? And then it went and found me a lactate dehydrogenase. And I was like, okay, this is cool. It is able to generate this new protein with certain properties that seem to have all these key sites that are reserved. And once they are conserved, you can actually generate the structure. So the purple structure is 5-MDH, which is a PDB structure of the wild type, which is from E. coli. And then you have the two orange and pink structures that were generated from the model. It so happens that they actually align really well with respect to the structure. They have very similar active sites, except for the fact that it might have a different substrate too, which we have not tested, but this is all pure hypothesis in that sense that we have these proteins. But the more challenging example is when I say, I'm going to only allocate 75% budget in terms of your sequence length, and then say, generate me a protein that is malate dehydrogenase. And it goes and does the same thing, right? It does its magic and it gives you this protein. And what I want you to observe is the green structure, which is the generated structure, preserves the catalytic site and the cofactor binding site, but it gets rid of a bunch of regions that it thinks are not essential for the catalytic activity. And so we were like, okay, this is fooling me. Really, this is not good. So we said, okay, let's run detailed simulations of this. We run a method called this empirical valence bond method, which is a semi-empirical method to basically test out if you can actually simulate an entire reaction step. And it so happens that the generated sequences that you see on this side, which is, I don't know if the boxes will come, but anyway, on this side, which is one of the sequences that we have generated here, it so happens that it actually has nearly 7 kcal per mole less activation energy than the wild type. And now this is kind of surprising because you don't expect a protein that is actually generated from scratch to have quite a lower height than what it is. And so we said, can we actually characterize at least a bunch of them that are not in the training data that have similar activation energies? And these activation energy profiles clearly show you that if you just take a look at the sequences that we generated using the reinforcement learning approach, it's able to generate those sequences that have better activity than the wild type. In this case, the wild type is malate dehydrogenase from E. coli. There might be other enzymes or other malate dehydrogenase that have similar function like what we have seen in the generated ones. We have not compared all of them because that would be another 700 comparisons. That's a lot of simulation budget that we cannot afford. So we have done this to basically say, okay, it does have better activation energy for malate dehydrogenase. From these samples, we have chosen another 30 of them and given it for wet lab experiments. So it is in the process of basically validating what we have in our simulations to say we have the same level of activity. So that's something that's going on. Now, moving on to the next portion, which is to say, can we use these models at the foundation level to predict SARS-CoV-2 evolution? This is actually the workflow that we had to develop to do this. So I talked to you about the foundation model. I also talked a little bit about the fine tuning on the SARS-CoV-2 open reading frames. But once you get these, you have to do this prediction workflow that allows you to basically generate new SARS-CoV-2 genomes and then check for their semantic similarity with the other genes that other genomes that are out there, and then look whether we can actually quantify the immune escape parameters or the fitness evaluation. That's all biophysical characterizations that are run using very expensive MD simulations. So we do have that as a full workflow now, so you can also use it. What is special about this model is the fact that we only presented the first year's worth of training data, right? We say, okay, I'm going to show you only alpha and beta strains, right? The rest of the data, which is about 6 million sequences that are out there, is all testing data. And if you end up doing that, the model actually presents very nice results, which is to say, okay, you can look at the alignment scores, they split up very nicely, but the alignment doesn't tell you what the sequence strains are. So you can now look at the strains also, and they categorize really well with respect to alpha, beta, delta, gamma, and so on. What is even more interesting is the fact that if you take a look at those individual islands that are present in the data, those islands corresponds to biogeography of where the viruses originated. Again, we did not give this information to the model. There's no annotation in the data that basically says this is from India, this is from US, this is from Canada, and so on. It's something that the model is picking up. I've not colored it with the examples from other countries, but here you can basically take it that these islands that we learn are coming from different pockets of the world. Oops, sorry. Can you go front? All right. Once you have that, you can also start generating new sequences, which is kind of cool because you now have the way to say, okay, I have these sequences that I start generating. The red colors are the ones that were generated. Gray represents the original phylogeny. And we can actually present results which are like 120 mutations from the place where we started, right? So in some cases, with respect to the Delta strain or the Omicron strain, we are actually like much farther in terms of our evolutionary trajectories. And these trajectories are sort of important because it also lets us identify at the time of November 2022, where PQ1 was one of the this ones that was actually gaining strength. We could predict with our workflow that one of those mutations actually represented BQ1, right? So this is without any knowledge to the model about the rest of the years of the trajectories. It was able to predict that there is one strain that is close to BQ1 in the generated trajectories that we had. So that raised important questions and said, okay, if I don't give you any more data, can we still predict EG5 characteristics, for example? And yes, the model does predict the presence of EG5 in the data. This is actually a MASH distance score, which is a whole genome comparison of where these different locations are. And you can see that there are two models that we are comparing here, depending on the level of generation identity that you want. And so it so happens that EG5 and XBB, although they have slightly higher MASH distances, they are present in the data, right? So this is kind of interesting that the model, without much supervision, is able to like predict new strains of viruses based on these phylogenetic analysis that you might traditionally end up doing. But the language model is somehow able to capture these characteristics of where the evolution might take it. Is it better fit? Is there better fitness? Is there better adaptation? I have no idea because we have not controlled it for generation in some sense, but it is having those ideas in terms of what it can learn. Right. So I gave you two topics that I talked about, the foundation model and the generative aspects of what happens with these foundation models. Let's talk a little bit about what we have been doing in the lab in terms of embodied agents, right? So within Argonne, we actually have this idea that we want to start with a system that provides a very high-level description of what the hypothesis looks like. And we want to be able to have the entire system be executed end-to-end autonomously without much human intervention. So this means that we need to have AI and robotics in the loop for planning, execution, and analysis of experiments. So this part of my talk is mostly going to talk about that aspect of what happens in the planning and running and all these things. What you see on the left-hand side of this is this virtual world implementation of what we are doing in the physical world, which is on the right-hand side. And I can give you points for, basically answering what that protocol is. It's one of the MolBio 101 protocols that you usually run. But to teach the machine to do this is a very different thing, right? So it means that it needs to understand what instruments it needs to bring together, what sort of things have to collaborate to actually form this, and where it needs to go and so on. So if you have more interest in this sort of work, I would highly recommend you can come and talk to me, because we have a bunch of programs that have now started for training both undergrads and grads in this area of like autonomous exploration or autonomous, discovery. And, an embodied agent in some sense is a computational system that interacts with its environments and learns from those interactions. So in the previous case where I showed you reinforcement learning based design of proteins and things like that, those approaches were mostly based on the fact that you had an environment of the latent space, and we are exploring that latent space based on experimental constraints. In this case, we are really asking you to explore what is happening within its natural environment. That is what you're seeing in the world. So a computational system has to be persistent or stateful. It manages the state information. It can sense and interact with what is happening outside and respond to those, right? This is physical or virtual, and then learn and adapt responses from that. So this idea of building embodied agents is actually not really that old. But, if you've played Minecraft, there is a recent paper from NVIDIA that showed that an AI model could actually learn new skills of all these things that you're seeing on the left-hand side of the screen, which includes hunting a pig, catching a fish, developing all the tools it needs to build for that, and all those actions that need to come out there, right? This is from a collaborator of mine, Anima Anand Kumar, who is at Caltech and who has been like developing these algorithms to do this. And there's always this joy of seeing how agents, gaming agents, can actually be built out of very simple descriptions of what the jobs can be. So in the case of actually like building these embodied agent, it so happens that when you consider AutoGPT or Reflection or React and any one of those systems, the Voyager actually tends to outperform all of them, where you have these new skills that it has added very quickly or new tools that it has added very quickly, and also mastered certain skills. And to get to that point, it actually had to devise a few things correctly. So it had to do this automatic curriculum thing, which means that, it has to discover as many diverse things as possible by just putting together something that's already present from the training data. You have to generate executable code for an embodied control, which means that you have to have an iterative prompting mechanism that allows you to generate any functions on demand. And all these things have to be stored and recalled with some sort of precision so you can actually run them at scale. So if you have any interest in like exploring this, is actually kind of cool. You can go and play the entire MindDojo game online, and you can sort of see what sort of actions it is taking and how it is using the environment feedback to like code these actions. So we said, is it possible for us to basically do this in the context of designing a science experiment, right? A scientific assistant of sorts. So you need to be able to configure and run computational simulations. You need to configure and run physical experiments. You need to collect data. You need to search the literature for data and protocols. You need to formulate hypotheses and so on and so forth. Everything that a grad student does. Does it mean that we're going to replace a grad student? No. But it's going to enable the grad student to be a better person in terms of learning the skills that they need and adapt them for new work environments, right? So unfortunately, this thing is though like fraught with a lot of challenges. So that if you have to act on resources, which include these local agents, you need to find varying interfaces, behaviors, all these things that you cannot really use very well. So if you use PubMed on a daily basis, I'm sure you would have faced this, issue, which is that you have multiple logins for the same app. And you don't know which app is using what authentication depending on, where you are in that whole story. So if you have to execute these actions reliably, you need these research services that need to run in terms of very scalable, facilities, but that's really not possible right now because you have all these failures that need to learn from the failures and do these things. And then, of course, without much, authentication and delegation is something that's very hard for these agents to do. And we don't have good ways to even think about it right now as far as building these agents. But what can we do without that? So in this case, we said, okay, the easiest thing for us to do is to design an antimicrobial peptide. And why is that? Because we had all the instruments available in the lab. We had a peptide synthesizer. We had the strains that we were working with, nearly 700 strains of bacteria. And we said, okay, if we want to generate something that is useful from the language models that we have learned, let's use this as a way to demonstrate that it's possible. So the challenge is to basically design an AMP or an antimicrobial peptide that can kill a specified bacterial strains without harming the host cells. But the problem now is that you with 20 amino acids that are there, you have 20 to the power of 20, which is 10 power 26 antimicrobial peptides of length 20. So this is a place where you can actually use the database to your advantage. There are millions of databases out there that talk about antimicrobial peptides. So you can adapt some knowledge from there and try to do this. And so we said, okay, let's engineer the platform to actually think about how to do that in the lab. So you have the peptide synthesizer that's located in the center. You get a set of vials that's basically the prepared peptides or the purified peptides. And you can basically run them through 7 strains of 96 peptides a day. Between 16 and 24 hours. That's because we need to give the bacteria enough time to grow. Otherwise, we cannot measure anything. And then you have this robotic screening platform that basically allows us to look for whether the bacteria have grown. And you do a very simple growth assay to calculate whether the bacteria died or survived. But you also have the simulations that capture successful and unsuccessful experiments, which can now be provided as input for our large language models. All the things that we did in terms of the reinforcement learning feedback, the thing that we are building in terms of all this was exactly for this reason. So we could actually provide all the feedback to the AI to constrain its generation, right? But you should observe that there's quite a bit of this throughput or bandwidth change that happens. So from thousands of peptides an hour, which is the language model that can generate it, it's only 96 peptides a day that we can generate. And then you only have 7 into 96 peptides that you can that you can screen in a given day. So there is a big gap there's a big gap in terms of how much simulations you can do, how much generation you can do. So that's something that we need to worry about when we are designing such experiments. But some questions that you need to ask your agent is, if you know that the peptide is a known antimicrobial, what is its most likely mechanism of action? All these questions are really available in the literature. Peptide X has some activity against G strain of E. coli. What is its main mechanism of action? So you're now refining what you got in Q1 to become more refined in Q2. And you can do all these things, but you need to have those protocols written up and do them. So an agent that actually does that would look up something like this, which is that you have the PubMed and ChatGPT, where you can query the PubMed for ChatGPT feedstock to basically get an insight into what questions are being asked. Then you have a separate agent that is called the UniProt agent, which aligns the proteins, predicts the structures, ranks the results, and says, okay, are these things doing the right things in terms of looking at the right structures that we want to play with. And then once you get that, you're now putting it into the BVBRC agent, which now answers questions related to what sort of strains might be the most responsive against these peptides that we have chosen. So each agent has a separate set of skills that it needs to master and then eventually come up with ways to run them, right? And now we actually drive that into a self-driving lab, right? So you can now take these peptides as input, chain them together using library services that are offered by Globus. If you have not heard of Globus, look it up. It's a cool service that's for basically data transfer around the world, but now it's being used as more compute resource for computing on these agents. And each of these agents has a set of roles that they play in terms of what queries they can answer and how they can answer. And then you take them for experimental evaluation and the self-driving platform that we have built inside the lab for like various tests. So your inputs is just a set of peptides. Your output is basically a set of validated scores that says whether it acts on a certain strain or bacteria or not. Because of a lack of time, I'm going to cover this mostly in terms of like growth assay applications. That's easy to do. We basically measure, at varying concentrations, whether that particular peptide can act in the lab. You get plots like these, which the agent can basically translate as actions for the next round. But you have to translate these lab protocols into a set of subtasks. And given the subtask, we can actually solve each task and recall them skills. So this agent is for automated lab code generation, which in this case happens to be an agent that takes in the task of interest, decomposes the task into a certain set of like subtasks, and then refines these skills on the fly when it is running on the lab. So the ability to run this in the lab while the execution is happening is kind of cool. And then it basically memorizes and keeps them as a list of tasks that it can recall whenever it needs to run them. Not going to give you a demo of how it works. I'm happy to like show it to you separately and how it learns to self-correct itself and then basically runs them in, tandem. So these are two agents that we talked about just now. But when you define these additional experiments based on the outputs that we get, we can actually run them at scale. Right. So this AMP designer is basically something that learned all these skills from the beginning. It has this HPC data repositories, robot instruments, and everything that it can program. And then eventually it leads us to basically build new AMPs from scratch by using various techniques to learn in the lab. I won't play this video, which is to say, what is our philosophy for thinking of scaling this to an entire facility, which is a smart science factory, because of lack of time. But I'm happy to show you how we have been using ChatGPT to come up with graphs of execution across the robots and then thinking of how to run them. This is a program that recently started from Argonne, which is to support the training of the next generation of workforce. So if you are interested, you can come and talk to me. There are lots of opportunities where we have, open lectures, Jupyter notebooks, interactive assignments, evaluation of these assignments also at scale, using all of the facilities that have been put together for, from Argonne for this particular type of work. This is mostly serving institutions with a certain level of, populations in R2 schools and things like that, but it's open for everybody. We are going to make everything available on YouTube and also on Jupyter Hub and things like that. So you're welcome to try it. With that, I want to thank all my collaborators, Anima in particular, for like bringing together a lot of the infrastructure and also the Minecraft story to us in terms of how we can use that. Cerebrus collaborators who actually worked on scaling these models. Of course, the lab entirely for a lot of the people that worked with me on the projects, visualization, data infrastructure, and also the self-driving labs, right? With that, I want to thank you all for your attention. I'm happy to take any questions.
Thank you so much. We've got time for a few questions. I know we have a break coming up, so if there are a few questions, we'll take them.
Yeah, please.
Yeah.
Maybe.
Andrew?
Thank you. That was a great talk. I'm really impressed with how far you guys have gone with agents. I have a ton of questions, which I'll ask you later on, but I just wanted to have a few high-level questions. One question is that you talked in your first project about SARS-CoV-2 with transformers about using reinforced learning with with feedback, we'll say. In this second project this project about antimicrobial peptides, is this also with feedback or?
Yeah, there is feedback in terms of controlling the generation of these peptides.
And my question is like in this kind of closed loop setting, how do you know the ground truth to give feedback here?
Yeah, that's a great question. And that goes back to validating with things that we know will really work in the lab. So for example, when we take these peptides, we don't make random modifications to them right now. we start with peptides with known antimicrobial properties across certain strains. And then we have generated a lot of training data for that sort of validated pairs that we know, but these are samples across the different portions of the landscape that we know we want to explore. So for now, the exploration process, we are basically limited to a very small number of mutations that are possible on the peptides that we work with. On the other hand, when we have these things running in the closed loop, the training itself can be an impediment in terms of running, which is where we are actually now asking Cerebrus or other instruments to be involved, where there is a component of AI at the edge to like push this together. So there's quite a few challenges there in terms of like addressing that question of saying, how do you get them to be the validated ones? But in this case, we are starting with some validation in the beginning. That is to seed the startup of our experiments. But then we are also seeing that when we generate these new examples, some of them have nothing to do with the parent seeds at all, which is kind of interesting and cool, I feel. But time will tell us. I think the aim is to try and see if we can decide an AMP within like 3 or 4 rounds of experimentation within 2 weeks. I don't know.
Got time for one question and then we're gonna.
Hello, that was a great talk. I had a question specifically on the 2D representation of the like high dimensional space, sequence space. Did you do any like representation functions, or did you just like grab the let's say the classification token, or did you grab like the padding of the entire embedding and throw it into like UMAP?
That's all we did.
Okay, cool.
Okay, I think you can go ahead for the break, and then Arvind will be available for additional questions. So we're going to convene back in like 8 minutes.
8 minutes? 10:20.
Who obtained his bachelor's degree in biology at Wuhan University. And I'm very proud to say that Wuhan University is the best university in China to study biology, because that's where I did my undergrad. And then Dr. Hu received his PhD in biostatistics from University of Pennsylvania. And right after his PhD, he joined Emory as a tenure-track assistant professor in the Department of Human Genetics. And right now his research group is focused on method development for single-cell and spatial omics data with a focus on Alzheimer's disease. Without further ado, we'll give you Dr. Hu.
Thank you so for the nice introduction. And today I'm going to talk about my work on statistics modeling and machine learning methods for spatial genomics data. But before we go to the key part of the project, let me talk a little bit about my research journey. I started my PhD at Penn in 2016. At that time, I joined the biostats department and working on electronic health records data on breast cancer. We did a lot of survival analysis. And after that 1 year, I was involved in RNA sequencing analysis. At that time, single-cell RNA sequencing is not popular. It's more like a bulk RNA sequencing. So we involved in a lot of projects like isoform detection and allele-specific expression. And after that year, in 2018, I switched it to single-cell genomics data using machine learning algorithms. So we developed methods for cell typing, cell clustering, and the differential expression analysis. And one components of our project is to apply this newly developed tool to different human diseases. So more recently, spatial transcriptomics is becoming more popular and mature. So that is a focus of my dissertation, to develop machine learning tools to analyze spatial transcriptomics data with different tasks on spatial domain detection and spatial variable gene detection. And after I joined Emory, my work is continued on spatial transcriptomics and on multi-omics data integration involving the proteomics, spatial metabolomics, and the genomics data analysis. So we try to develop these methods and analyzing data from tumor and cancer and Alzheimer's disease, trying to solve different disease diagnosis and help clinicians and physicians in their own research. So I established the AI in genomics in Emory last year. So you can tell from the name, it's pretty direct. We work on genomics problems and using AI tools. So this is a question always people ask me is like, what is AI and what is their relationship with the machine learning and statistics? What are the three parts? So my own understanding, usually people talk about AI, so they talk about three parts. AI has three parts. The first part is data collection. You need to have large type of data, and then you can to do any prediction on that. And the second important components of the AI is algorithm itself. You need to develop a model or develop a statistical analytical tools to utilize this data. And the third part of the AI part is the computational resources. Usually students complain to me like one model may run for a few weeks. So computational resources, especially the GPU or the chip CPU, they are very important. But for my lab, we work on algorithm design. So in this time, algorithm, it also have a different aspect. Usually algorithm, so it's never, it's not something new that explored and get popular these years. Algorithm and AI parts is something started from a long time ago, just becoming popular in these years. So algorithms start from the statistical tools, like linear regression or this type of thing. They are analytical tools, so they are called algorithms. And more recently, machine learning, deep learning, this newly developed tool are getting more popularity from people. So they all fall in this part as algorithms. So my lab works on developing algorithms for genomics data. So before we get to how we tackle different problems, let me give a brief introduction of the spatial transcriptomics data. It's a very popular technique. So spatial transcriptomics data is a technique which can measure the gene expression of a specific tissue while also preserve their spatial location information. There are a few platforms which get commercialized and are well adopted by the community. I would categorize the spatial transcriptomics technique into two types. On the left hand side, you can see it's called the Visium system, which is a sequencing based, which means it's an advanced version of the single cell RNA sequencing. They can sequence all the spots, I would say, and then put their spatial location information in the whole tissue. And the second category of the spatial transcriptomics is the hybridization based or image based, which include the merge scope technique and Xenium technique. So these two types of technique, they differ from two aspects. First is for Visium, like the sequencing-based technique, usually they can cover the whole genome, which means they can measure all the genes in an unbiased way. But the drawback of this technique is that the resolution, they cannot reach to single cell resolution. Usually they are measuring the gene expression information of a small box, which usually contain 5 to 10 cells. So in the contrast, the imaging-based and hybridization-based methods like a merge scope and Xenium, they can measure the gene expression at a super high resolution, even subcellular resolution. Like you can identify different cell types and map their expression level. But a drawback compared to Visium is that they can only measure a limited number of genes, usually 300 to 500. So compared to Visium, maybe merge scope and Xenium, this type of technique is more suitable for validation. For example, you are trying to do some experiments and you already know what genes you want to look at with a pre-designed panel, you will choose merge scope or Xenium. But if you want to do some discovery study, like unbiasedly check the microenvironment of some specific tissue, you don't have a target of the gene panel. So you can try some technique like Visium. As I know that the Visium technique and merge scope, they are definitely one biggest challenging part of their technique is they are trying to increase the number of genes. And to my knowledge, maybe next year, they can increase the number of genes from 300 to 1,000, but it's still compared to Visium, which can measure the whole genome containing more than 10,000 genes, it's still a big drawback. So let's take a look at the data we get. What the data looks like as a spatial transcriptomics. This is a mouse brain data, and on the very left, you can see a H&E image staining of the whole mouse brain tissue. And for the gene expression data, it's presented as an expression matrix. And in this matrix, each row corresponds to a spot, and each column corresponds to a gene. And the number in this matrix represents the UMI count, like the gene expression level. And you can see as here, I term them as spots because one spot here, usually in Visium, they have different size, like 50 or 30 micrometers. Each spot can contain multiple cells, and these cells, they are not necessarily coming from the same cell type. And spatial transcriptomics, we need to have spatial information. So in addition to the gene expression information, we also have these two columns, X, Y. These two coordinates, 2D coordinates, can map each spot back onto the histology image. So we know where these spots are located. We can study the microenvironment, their neighborhood information in a very efficient way. So with this type of data, there are many questions people are trying to ask. First type of question is spatial clustering. So spatial clustering is a statistical term, but the spatial clustering, the goal of it is trying to separate the whole region, whole tissue into different subregions. For example, in brain, you have cortex layers, white matter, gray matter. How can we separate them? And then we can examine the gene expression pattern at each region individually. And the second part is that in single cell RNA sequencing data analysis, usually people sequence all the single cells and they are trying to ask, what is the cell type markers? What are these cells? So they are trying to find cell type markers. But in spatial transcriptomic data, we do not have single cell resolution on Visium, but we can investigate on what are the genes that show some spatial variability. As shown here, some genes will show a specific spatial pattern, which has a biological meaning. So people are trying to identify spatially available genes rather than looking at the cell types. And as I said, with spots do not have single cell resolution. So deconvolution is another task which people are trying to solve. Then after deconvolution, we will know for specific cell type, what are their spatial distribution at different regions, and then they can do some investigation on that. And another advantage of this technique is that it allows us to study the cell-cell communication at a spatial level, because usually for single cell, when you infer cell-cell interaction, you just look at their expression. But you know, two cells need to interact when they are close enough to each other. So with the spatial information, we can get more information and constrain this type of cell-cell interaction analysis in a specific location, which give you more precise and accurate inference of the cell-cell interaction. So the first part of the talk is to focus on the project of the task of spatial domain detection and the spatially variable gene detection. So imagine that in spatial so before we do spatial domain detection, what is the concept we should define? What are spatial domains? So spatial domains here are defined as regions that are spatially coherent, both in gene expression and their histology image. That means they need to show similar gene expression and they should be located together. Then we can identify them as spatial domains. And why we are interested in identifying spatial domains is because it can help us to understand how the gene expression of cells is influenced by its surrounding environment and how neighborhood regions interact at the gene expression level. So given the data structure we have, in this task, what type of information we can utilize? First is definitely most important, gene expression. And second is the spatial location is also very important. And third, usually for spatial transcriptomic data, usually for Visium and Murphy's, they have a HNA staining image. So we also have the cell morphology from the HNA image. So there are 3 levels of information we can use, and each of them only paint an incomplete picture of the whole task. We want to combine all of these 3 modalities together. So to solve this goal, we develop our own method called Jian. The goal of Jian is trying to identify spatial domains by spatial clustering, utilizing the 3 levels of information I just mentioned. And after the spatial regions are identified, the next question people will ask is, what are these spatial regions? Are they white matter or cortex or tumor region or lymphocytes? So we want to identify genes that are highly enriched in each spatial region. We call them spatially variable genes. These spatially variable genes will uncover the biological function of each spatial domain. So next we go to the methodology part, the algorithm part, the fun parts that is difficult to understand. So the key idea of this project is that we have 3 levels of information: the gene expression in a matrix, the spatial location in 2D XY coordinates, and we also have a HNA staining image as an image format. Then how can we combine these different information together as they are in different data structure? And our answer is that we want to utilize an undirected weighted graph. So as you can see here, on this undirected weighted graph, each vertex, which means each node here, is a spot with their gene expression information as their node features. And then we connect this graph using edges. And weight on the edge is determined by 2 information. First is their spatial location, and second is their histology image color. So to be more specific, we can calculate the Euclidean distance between any 2 spots. So instead of using only XY, their spatial location information, we'll also add the 3rd dimension Z here. So this Z represents the color intensity on the HNA image. So you can see, as shown in this image here, darker the region, higher the Z color. So by adding this Z color, 2 spots are considered to be close using 2 criteria. First, they are spatially close. Second is they have similar color on the HNA image. And then we convert this distance into a weighted weight on the edge to make sure that longer the distance, smaller the weight, because weight kind of measures the similarity between two spots. So for any tissue section, we can build this graph. And after we get this graph, how can we put it into a machine learning model? We use some specific structure called a graph convolutional layer. So this graph convolution layer is used to extract information from the constructed graph. And then we use the iteratively transfer learning methods to identify the spatial domains. So because the way we integrate the three levels of information together, the spatial domain we detected should show similar gene expression, spatial coherence, and a similar pattern on the H&E image. And after the spatial region identified, we are trying to identify like genes highly enriched in each region. For example, here, we identify an orange region, and we identify genes that are highly enriched here. We also find that for some region, maybe one single gene is not enough to mark one region. So we have another algorithm to identify a linear combination of multiple genes and then combine them into a meta gene. And this meta gene will mark this region. And if you look into the detail of the composition of this meta gene, it will kind of tells you what are the function of this region. So that's all for the difficult methods part. And then we go to the application part. We first apply our methods to a mouse posterior brain data. As shown on the left region, it's the histology image with very clear pattern on the white matter, and on the bottom left is the top left is the cortex layer. And by applying our methods, we can successfully separate the whole tissue region into different domains. And next is we want to identify domain enriched genes. So for domain 1 here, for example, we identify this PVALB, which is highly expressed in this region in domain 1. And the TRM62 is highly expressed in domain 8. So you can see, although domain 1 and domain 8, they are spatially coherent to each other, these two genes can perfectly separate and tell us about their function. And for domain 5, we identify this NRGN, which is highly enriched in the brain cortex. An interesting finding that this NRGN is also highly expressed in domain 7, which is located on the bottom left region. So this gives us some understanding of maybe these two regions, they are neuroautomatically similar. Both of them consist of the cortex and the pyramidal layers of the hippocampus. And they are just separated artificially because of how the tissue section was cut. And these two regions, they are highly, this domain 5 and domain 7, they are both enriched for the cortex and the hippocampus. And they are, by a 3D reconstruction, they should be located on the curved surface of a brain, so they may be continuous. Another example I want to show you is for the domain 0 here. We find that it is difficult to use single gene to identify this domain's identity. So our methods will give you a linear combination of multiple genes. So for example, by starting from this KLK6, which is highly expressing part of the region in domain 0, and adding this MBP and subtracting this ATP1B1, we will arrive at a single meta gene, which perfectly marks this domain 0. So then we look at the function of these three genes. In this meta gene, we consider the KLK6 and MBP as positive genes because they are highly expressed in domain 0, but they may have some noisy pattern in other regions. While this ATP1B1 is considered as the negative marker because it does not express as domain 0, but highly expressed in other regions. And then we find that this KLK6 and MBP, their expression are restricted to oligodendrocyte, while this ATP1B1 is mainly expressing neurons and astrocytes. As this reasoning that affects domain 0 represent the white matter, which is dominated by oligodendrocyte and have fewer neuron cell bodies. So the components of this meta marker will give you some understanding of the spatial region. So the next thing we are trying to do is that if we have multiple tissue sections, for example, if a mouse brain is too big, we cannot fit it into one slide. Can we do the mouse brains, cut it in multiple slides, and then combine them together? And the answer is definitely yes. So on the left is the mouse anterior, on the right is the mouse posterior region. We just jointly analyze them together, and our methods is able to identify the shared region, the cortex layers, for example, the yellow, purple, and the brown parts. And for anterior and the posterior, they also have their own unique region. So it's pretty accurate. If you have multiple tissue sections or a larger tissue section, you want to cut them into multiple sections, we can combine them together. We also try our methods on a pancreatic cancer tissue. And on the left is the histology image with manual annotation separating the whole tissue region into tumor versus non-tumor. And our methods is able to identify domain 2, which is colored in black here, correspond to the tumor region. But based on this result, we see that the resolution of the spatial transitomic data for Visium is still pretty much low. We do not go to the single cell resolution. And based on this result, one question I ask myself is that, can we get more detailed information of the tumor microenvironment rather than just separating into tumor and non-tumor? Can we get more detailed information? The answer is yes. So that's why I developed another tool called this Tesla, short for deciphering tumor microenvironment at a super resolution using spatial transcriptomics data. So first is some background knowledge of why we care about the tumor microenvironment. So first is, what is tumor microenvironment? Tumor microenvironment usually refers to the tumor and its surrounding area, which consists of the tumor cell, tumor stroma cell, immune cell, excellular matrix, and different signaling molecules. People care about the tumor microenvironment because it is related to the occurrence, growth, and metastasis of the tumor, and it can give you some hints on the treatment plan choice. As shown here, tumor microenvironment is a very complex region. And usually if you look at the Visium data, the original Visium data, each spot contains multiple cells. These cells, they are highly possible came from different cell types. So only separated this domain will not get enough information of the whole tumor microenvironment. Another information we find that the histology information is very useful. As you can still remember, in our previous methods, we only use the color intensity as the Z value. But in fact, the histology information will provide us more rich information. For example, these are three subsets of one H&E image. And you can see their cells nuclei shape, size, and density are very different. And the well-trained pathologist can tell you what cell types they are from this histology image. But we also have to admit that for some of the cells, for example, the subset of T cell or B cells, these lymphocytes, they look exactly similar on the H&E image. So we still need gene expression information further to distinguish these cells subtypes. So this example just shows that the gene expression and the histology image, they are complementary to each other. Each of them tell part of the story. We need to combine them together so we have a whole picture of the tumor microenvironment. But before we combine the gene expression and the image information together, there are a few challenges we need to solve. First is the difference of the resolution. As I said, the H&E image can be super high resolution. They have 20x or 40x with the whole skin of the whole tumor region. But for spatial transcriptomics data, first is the resolution is pretty low. It's 55 micrometers, and we only have non-single cell resolution. Another thing that the measured spots here, as shown here, no matter which technique you use, the measured spots are far away from each other. So they have tissue gaps between these spots. We need to fill this gap before we integrate them with the H&E image. So before we combine them, we first need to solve the low resolution issue, and we also need to fill the tissue gaps, which are not measured by the spatial transcriptomics data. So to do that, we develop our methods for tumor edge structure and the lymphocyte multi-level annotation, short for Tesla. The goal of Tesla are threefold. First is we want to enhance the gene expression information's resolution to the same as the H&E image. As shown here, on the left is the gene expression, original gene expression, and after enhancement, we will get a super resolution gene expression here. And after we get this gene expression image, it's the same format and the same resolution as the H&E image. Then we can combine them together to annotate the tumor region, to annotate the lymphocytes, and then we can characterize the intratumor heterogeneity at a super high resolution, which cannot be achieved by our previous method, just separating different regions. So to do so, the very first step is that we start from the H&E image, and then we scan the contour of the whole tissue, and then we use grid to separate the whole tissue into small boxes, which we call these small boxes superpixels. The size of each superpixel is much smaller than the measured spots. And then we put all the superpixels and the measured spots onto the shared three-dimensional space. The two dimensions are X and Y, and the third dimension Z here is the color intensity. As you can show here, each small boxes is a superpixel, and the circles here are the measured spots. And then we'll calculate the nearest neighbor, and based on their distance, we can impute the gene expression of each superpixel. And after all these processes, we achieve this super resolution gene expression. So after this step, we can see all the genes are converted from the expression matrix into images, and then we can combine the images with the H&E image together. So usually for image, we have three channels, red, green, and blue RGB. So right now we have a high dimensional image. Each channel is a gene. So for example, we have 100 genes and the H&E image, so we will have 103 channels with high dimensional images. And then we can use some machine learning model like a convolutional neural network, which is designed for image data analysis, and then combine them together to do image segmentation. And based on the image segmentation, we will annotate the tumor region. So this framework is very flexible if you change the tumor marker genes into the cell type marker genes. So you have a different high dimensional image and then do the cell segmentation, we will get different cell type distribution image on the whole tissue section. So this is kind of the convolutional neural network we use. So the key idea of this whole neural network is that we're trying to treat gene expression as images and then stack them with the H&E image together. And then we can use some well-defined methods in computer vision methods to do this type of deconvolution and get tumor region or different cell types region annotation. So here are some applications to the cutaneous squamous cell carcinoma tissue. On the left is the H&E image, and in the middle is the tumor region we identify. So our methods can also identify the boundary region of tumor, which we call tumor edge. People care about tumor edge because usually tumor edge is more active than the tumor core. They are trying to invade into the normal region, so people want to draw the tumor edge. And this is the manual annotation of the leading edge of the tumor, and our methods is able to draw the detailed structure of the tumor edge. Second part, as I said, one goal of Tesla is that we have super high resolution annotation of the tumor, and can we characterize the tumor heterogeneity? That is, we separate the tumor into tumor core and tumor edge. As shown here, after we identify the tumor region, our methods can still separate the tumor into core, which is colored in black, and edge region colored in red. And next part is that we want to identify genes that are highly expressed in tumor core edge. This gives you some understanding of the tumor's situation. For example, we identify core enriched gene, this IGF-BP2 and CCMB1, they are highly enriched in tumor core. And this CXCL12 and IR2RB are highly enriched in tumor edge, only in the bottom region. So that means the tumor edge on the bottom region are pretty much activated. These two genes are related to the activation of CD8 T cells. So that means there is anti-tumor immune response happening at the bottom of the tumor. It's more active at the other side of the tumor. It gives you some understanding of how the tumor is invading into the normal tissue. So next, by changing the tumor markers into cell type markers, we can identify different lymphocytes on this whole tissue region. As you can see here in different color, we can identify B cell, CD4 T cell, CD8 T cell, and dendritic cells. All these cells have different locations. So next question we ask is that, can we identify some specific immune structure based on the colocalization or overlaps of these different immune structures? So one thing we want to identify is to identify the TLS, tertiary lymphocyte structure. So people care about TLS because it is related to the prolonged patient survival. Usually TLS structure is generated inside or very close to the tumor. So you can see its structure is signatured by the B cell surrounded by T cell. So then our methods allow us to examine the colocalization of B cell and CD8 T cell. We can see that they are highly overlapped at the region which is circled by the yellow boxes here. So our method is able to give you a TLS score, which tells you what are the potential region of the TLS. And these TLS can be further validated by examination of the pathologist. So in addition to that data set, we also apply our methods to a malignant melanoma tissue. Similarly, on an H&E image, we have manual annotation separating the whole tissue into melanoma, stroma, and lymphoid tissue. And follow the same pipeline, our methods is able to identify the tumor edge and the core, and identify core and edge enriched genes. But interestingly, for this data set, we find that the core enriched gene, this HSPB1 and MRF, they are hypoxia-related genes. So that means at the tumor core region, there is a lack of oxygen. And for the hypoxia-related tumor subtype, usually many treatments are will not be efficient because the hypoxia will hinder the delivery of different tumor agents. You may choose different treatment plans. And also for tumor edge enriched gene, we identify this ATGAL and the THLI1, which are all related to the activation of different lymphocyte immune cells. And we also identify different the TLS score, which tells you where are the TLS regions. So in this data set, we identify 3 suspicious regions. In the blue box and the yellow box, by further confirmation by the pathologist, we saw their true TLS region. But on the green box, as shown on the top right region, it's not TLS. Although we observed the aggregation of B cell and T cell, it is a lymphoid node rather than TLS because this aggregation is far away from the tumor itself. Usually TLS are deeply embedded inside the tumor region. So that is for the second project I want to introduce. And the third part I want to talk about is one of my ongoing projects. It's trying to navigate the molecular landscape of cell morphology changes in multi-omics data analysis. As you can see, most of the data used for like the Tesla methods we just published, they're trying to use some deep neural network, convolutional neural network to integrate the histology image and the gene expression information together. One advantage of these methods is it's pretty efficient. It is good at accomplish different type of job. But one drawbacks, I think most of you have heard, neural networks are black box, or at least a gray box. So you don't know what is the association between the cell morphology and the gene expression and how they affect each other. So as in our methods, we use convolutional neural network, and there are other methods like the Muse published on Nature Biotech. You can see this is a picture of their workflow. They use the autoencoder, some type of deep neural networks to do the embedding, and then combine them together to do spatial region detection. So the common thing they share is that, including the SIGRA method, they also use the autoencoder to combine cell morphology and gene expression together. The common thing they share is that all these models are black box model. It's pretty efficient, but you don't know how the gene expression change or affect the cell morphology change. So that's why we want to develop our own methods. And another fact is that usually, sometimes I just said the histology and the cell and the gene expression, they tell you the same story. But in many cases, we find that spatial domains are inferred by cell morphology and inferred by gene expression. They are not the same as each other. For example, maybe the pathologist will annotate the H&E image based on the cell's morphologies, and it's more related to autonomy structure, while spatial domain detection detect regions that share the similar function. They are not necessarily the same. They have some contradictory parts. But this method, since they use black box models, they are not able to tell which part are complementary, which part are conflict to each other. So we developed our new tool, and the aim of this study is to understand the relationship between the morphology and the molecular profiles in a transparent way. Most of the methods use black box neural networks and makes the extracted features hard to interpret and suffer from a lack of biological meaning. And many machine learning models, as I said, they need large data set for training. Usually these training data sets need a lot of labeling from pathologists. Sometimes it's not available. So that's why we identify our own methods called interpretable morphology features from unsupervised segmentation, short for MUS. So MUS is able to extract interpretable, meaningful, and robust morphology features from the images, and then quantify the association between the cell morphology change with the gene expression change together. So you have a better understanding of the whole tissue section. So the workflow of MUS, it starts from the H&E image by separating them into different patches and perform unsupervised segmentation. So you can see the segmentation results presented in mask mean different things. For example, the first mask, mask 1, represent the nuclei, and the mask 2 represent the stroma region. And then we can do objective detection to identify different nuclei individuals and the stroma connected regions, and do different measurements. For example, the first set measures the area of the nuclei, the dispersion, and the nearest neighboring site. And the second set of features measures each nuclei, what is the size, solidity, roundness, width, and height ratio of all this information. And after measuring all these cell morphology informations, we're trying to quantify the association of this cell morphology with the gene expression information, the spatial gene expression information. So some detail of how we quantify the information is that we dissect the tissue into different regions, and then for each feature, we separate them into XY two orthogonal directions, and then use variance-based correlation metric to quantify their 2D pattern similarity. You cannot directly use some statistical tool which do not consider their spatial information. So by separating the whole features into different sets, we can separate, for example, for gene expression, we can separate in 2 sets. First set is the gene expression feature which has no image feature similar to them. That means these features capture the unique pattern in gene expression. And for image features, similarly, we will have another set of image features which has no similarity compared to gene expression. These image features measures their unique pattern. But the interesting part is the intersection part. This part, some gene expression have the shared pattern with the histology image features. That means the gene expression and the cell morphology change on the same direction. It tells you how they affect each other. So here I'm going to show you some example with our application of bladder cancer sample. On the left is the whole tissue region, and we only focus on the tumor region, which is annotated here in red. So within the tumor epithelial region, by gene expression, we can separate the tumor into different 3 subtypes here. And we find that this purple subtype in the tumor region have very distinct gene expression compared to its surrounding regions. For example, we identify this B2M and this CD74, their expression are highly enriched only in the purple tumor region, even compared to its surrounding regions. And next, we identify some of the image features that are highly expressed also in this tumor, in this purple region. And then we look at what this feature is really capturing. So this feature captures the nuclei change. For example, from smaller value to larger value, it captures how the nuclei become bigger and its color becoming more light and darker region has more variability in its color pixels. So here, as we know that they are all tumor cells, and if a cell's nuclei is becoming larger and show variability in their color, it means it's more activated. The chromosome is opening and trying to transcript more RNAs. So this change is associated with the B2M and CD74, these 2 genes. So it kind of give you an understanding of how the gene expression change along with the cell morphology change and how they affect each other. Another example I want to show you is on the lymphocytes region. So on this lymphocytes region, we have 2 structures, TLS and TLL. So biologically, they are all aggregation of B cell, T cell, but TLS, these B cell and T cell, they are trying to aggregate and form some specific structure, while the diffuse TLL is just the immune cells, but they are kind of loosely distributed and do not form some specific immune structure. So based on the manual annotation, we can, from this tissue section, we have both TLS and diffuse TLL. And then we find that this MS4A1, which is a marker of B cell, is highly expressed in the TLS region. And we find that there are some of our morphology features can better capture the pattern. And this image captures the pattern of the cell nucleus. For example, you can show here. On the TLS region, the nuclei are more aggregated together with each other, while on the diffused TLL, all the nuclei are far distributed from each other, are more loosely distributed. So this image feature tells you the structure difference in the TLS and the diffused TLL and how they're affected by the gene expression itself. So the goal of our tool is trying to bridge this gap, bridge the cell morphology and their gene expression. And at last, I want to talk a little bit about my lab, AI in Genomics lab. So from my own experience, I have a lot of work on the multi-omics data analysis, including DNA, RNA, protein, and metabolomics. And this kind of like represent the genotype of a specific tissue. On the other hand, the image data, medical image data, including H&E, trachoma, MRI, all these image data more represent the phenotype of the different disease samples. So the mission of our lab is to develop statistical tools in an integrative manner to combine all this data together to fill the gap from genotype to phenotype. And then if we combine clinical information, we can use our model to do some early diagnosis, prognosis, and patient survival prediction, which have final effects on the real applications. So that is the goal of my lab. And that's pretty much all for what I want to share. I'll take time for questions. Yes.
Thanks for the interesting talk. I had just one technical question and one larger question. So you had in your graph convolutional network that you had, I think, decayed the edges by like an exponential, right?
Yes.
What's the biological justification for that?
Okay, so we use the extension decay because for 2 spots, we calculate distance. So its distance, the edge weight kind of measures the similarity. So if they're more similar, the weight should be larger. But the distance is that if the distance is larger, it means it should not be that similar. So the exponential decay is just to transform the distance to make sure the longer the distance, the shorter, the larger the edge weight.
So when you build this like induced graph, do you have like a cutoff based on distance or like a maximum number of neighbors or something?
Yeah, I know there are many methods. They have a cutoff. For example, for one node, they only select its nearest neighbor, right? But I mean, for the exponential decay, that means that if the distance is longer, it's trying to decrease in a very fast speed. So we find that if you use a cutoff or do not use a cutoff, it will not affect the model performance very much, because even you have a lot of neighbors, only the few neighbors will dominate the whole graph.
Yeah, it's interesting. We went through the same journey in chemistry. Basically, people arguing about, exponential cutoffs, having attention, learnable cutoffs. And most people in the end just realized it didn't really matter, and then they just let it train without an exponential decay. I don't know that chemistry is different than biology.
I think they're similar, because graph convolution is kind of a tension. It will automatically adjust for the weights.
Yeah. And my other question is, I didn't really get it in the first part of your talk, is that, with the autoencoders, with the self-supervised learning, it kind of makes sense. You have to learn some compressed representation. But in your earlier work, you would show these like class numbers, and they would be an integer between 5 and 25. Like, is there some ground truth, or how do you train that to give you the right number of classes, and how do you check that they're correctly assigned?
Yeah, this is a great question. So this is a general question for clustering, like K-mean. You can set K to be 10 or 100, but there are some, even you don't know the ground truth, there are some way to assess the quality of the cluster. For example, the silhouette score or intercluster ICC number. They are trying to evaluate within your cluster how homogeneous they are, like the intercluster and the between cluster difference. They can keep this balance. So usually when you're trying to decide the number of clusters, you try different numbers and you calculate these statistical numbers and you pick the optimal.
Okay, I just was hoping that you were going to tell me there's some, ground truth in genomics about the number of cell
No,
types or something.
no ground truth.
Unfortunately, okay.
Yeah, so for genomics, the ground truth is still pathologist annotation.
Very nice talk. Thank you. For your spatially resolved transcriptomics, how do you deal with biological replicates? And I guess I'm thinking specifically of if I guess if you're looking at a tumor, then that's a specific thing. But if you're just saying looking at developmental changes over time and you want to get significance of gene expression changes, how do you do that?
Yeah, this is a great question, because at the very beginning of this field, people are generating single section. But as this cost becoming decreased and people are trying to generate multiple sections, and they generate it in two different ways. First is we have one section, but we do consecutive cuts, and then we can reconstruct the 3D structure. Another study design is that they have different, for example, we study Alzheimer's disease, we have mouse at different months, and then we cut mouse brain at different months, and we have a time series data, and then combine them together. So batch effects, the batch effects is always exist, but usually we find that for some study, if they generate from the data generated from the same lab, same study, usually the batch effect is minimal. But if you want to combine, for example, for one data, you have mouse from 1 month. For another study, you have mouse from 6 months, and then you're trying to combine them together. Even they are the same tissue, there are some very different batch effect, even they are using the same platform. And also, as I said, spatial transcriptomics has different platforms. Even you generate the same sample by using different platforms, they have some difference. For example, the sequencing depths. For some techniques have pretty high sequence depth. That means they can identify a lot of genes that are lowly expressed. But for some other technique, they can only they are all zeros. So batch effect, the platform effect is always an issue here, and people are trying to solve it in a statistical manner.
Okay, I guess, I mean, it makes sense that within a sample, you can, that I can believe that you could easily compare across within a sample, but it just, even if they're the same age, just taking, say, mice that are reared identically, and you look at the gene expression between mice, that still sounds like it's a challenge, but I guess that's looking from one mouse to another mouse, even if their timing is the same.
Yeah, so in one of my recent projects, we have mouse from different time stage, and we're trying to quantify how their neuron cells change at different stages. So when we design the gene panel, we usually want to get some spiking gene or housekeeping genes. So we use this gene as anchors, and then trying to use this gene to adjust the expression magnitude across different time and ages, and then do the comparison. So it's challenging, but there's always some solutions in statisticals.
Hey, that was a really awesome talk. I just had a question about calculating like the metagenes and how many genes you can pack into each graph, and if you use like multiple graphs. So like when you are trying to like understand the multiple genes that go into creating these tumors, do you just use multiple graphs, or do you just keep appending different genes to like the meta gene function, if that makes sense?
Yeah, this is a great question. So we usually start from one gene, and then we try to add or minus some of the genes. And in each round, we have some stopping criteria. For example, if we have a criteria to check if the pattern is strong enough, so it will automatically stop. Some metagene contain 3, some metagene contain a longer list.
It was a good talk. And my question is that when you make the spatial domains, like if you just take the gene expression data by cells and then make the clustering in the UMAP, then how much it was corresponding to each other, to your spatial domains, to the UMAPs, the clusterings by only by gene expression data?
Okay, so we have tried this because UMAP is something you need to you use for visualization for single cell data. But right now, since you have XY coordinates, people are not going to use UMAP for visualization. And if you do not consider spatial information, for example, you only use gene expression to do the clustering, we will see some of the clusters are very sparsely or evenly distributed across the whole space, which means they may be dominated by some cell type, and this cell type is sparsely distributed across the whole region. So it depends on the purpose. If you want to identify domains that are spatially coherent, you should definitely consider the location information. And for some single cell resolution data, we usually do this in parallel. So that means we identify spatial domains. We also do cell type detection. And if you plot the spatial domains, you will see different layers, structures, autonomy structures. And if you plot their identity by cell type, you will see very messy mapping of the cells across all the space.
I can ask a question about the third part. So
Thank you. So for the third part, if I understand correctly, you learn the cell morphology from the historical image. So if you do not have those data, if you only have the spatial omics data, can you learn the morphology only from that data to see the size of the nucleus or where the size of the, especially for neuron, like in the brain, you have different cell shape. Can you learn those cell morphology from spatial omics data alone?
Yeah, so this is a very interesting question. And in this field, people are trying to do 2 types of work. First is, if they only have H&E, can we predict their gene expression from the histology image? Second way that if they have the gene expression only, can they redraw the H&E image? So H&E image is much cheaper than the spatial genomics. So usually people do it in the inverse way. They want to predict the gene expression based on the H&E. They are not trying to predict their H&E based on the gene expression because they can do the staining in a pretty cheap price. So there are some work on that. And if you're interested, I can send you a few ongoing works. People are trying to tackle this issue.
I think there's an interest in understanding the distribution of, for example, of a specific gene. Is it more in the nucleus or is it more in the cell plasma?
People are trying to do that, but it's difficult because we have single cell resolution, but you zoom in, you want to get a subcellular resolution. There are more noise, and sometimes it's difficult to do. And it's RNA, it's not protein. So people doubt the meaning of like RNA should be highly enriched in the nucleus or cytoplasma. What is the meaning of it? So there are some ongoing research on that, but there's no very concrete conclusion on this part.
Thank you. If there's no more questions, let's thank Dr. Jian.
Thank you.
Pleasure to introduce our first speaker and our very own Dr. Adrienne Hoarfrost from the Department of Marine Science. And Dr. Hoarfrost got her bachelor's degree in biology from Dartmouth. And then she received her PhD in marine science from UNC Chapel Hill. And she did her postdoc at NASA. And now we are very excited to help her here being an assistant professor in the Department of Marine Science. And I think soon she will join the Institute of Bioinformatics. And she's also a fellow in the Institute for AI. And we'll let her share more about her research.
Thanks so much, everybody. It's a pleasure to be here, and I'm really excited to see so much interest in AI and bioinformatics and biology. It felt for a long time, especially in my field of marine sciences, that I was kind of screaming into the void a little bit, and I think that's very much not the case anymore. So it's really exciting to see so much energy around it. So I joined UGA just last year. I just celebrated my 1-year anniversary as an assistant professor. So getting things rolling, and I wanted to share today some of my work in thinking about applying AI to environmental systems and environmental microbiology sort of writ large, but marine sciences specifically. If my slides will advance. There we go. My work has always been on looking into microbial communities in the ocean and how they impact the carbon cycle specifically, and biogeochemistry sort of more generally. So my early work was very much related to going out into the ocean on these big boats, whether that's on coastal ecosystems or the open ocean or down to hydrothermal vents, and taking your very precious samples, getting your water, getting your sediment, and running a bunch of experiments on it, trying to see what microbes are eating, what kind of carbon compounds are being degraded, and what that might mean for the larger carbon cycle. And the thing that I like to highlight for my AI-related talks is that this is really precious data. I mean, measuring anything in the ocean is really hard. It's very remote location. It's very extreme environment. It costs, maybe $40,000 a day to operate these boats that you go on. So they're very precious data, and you go out and you collect everything you can, and you wring as much value out of it that you possibly can. But most biological data in general, and marine science data specifically, is definitely small data. So when you're thinking about applying AI to all of these systems, it's always thinking about how do I leverage all of the biological data that we have to get insights into my very specific, very small data set. So I wanted to talk a little bit about just overview of the marine carbon cycle and why it's so important and why we care about this in the first place. So this is a very simplistic view of carbon cycling in the open ocean. But one of the things that I think maybe people don't appreciate, you look out at the ocean, you're on a boat and you see a bunch of water. It doesn't look like all that much is there. But in fact, in about a milliliter of seawater, there's 1 million cells. And so, half of the photosynthesis that's happening on Earth is happening in the oceans by phytoplankton living in this seawater. And they're microscopic, you can't see them, but they do as much photosynthesis as all of the trees and plants around us. And then all that carbon that's fixed in the surface ocean, some of that, of course, gets recycled by heterotrophic organisms, mainly heterotrophic microbes, and it's variable. It's extremely variable depending on where you are in the ocean, how much of that primary productivity, that photosynthesis, is getting recycled by heterotrophs. And this can be anything from, so what this figure shows is the export ratio, so the proportion of primary productivity that's being exported to deep water, and that ranges in this figure from about 0% to about 100%. So it's a very wide variability, and we're really bad at modeling that. But it's extremely important for the net carbon cycle and for climate, because that carbon export that's biologically mediated, we call it the biological carbon pump, and it's the single largest net flux of carbon out of the atmosphere, period. Not biological carbon, just carbon. And once it gets into the deep ocean, because of the way that ocean currents work, it'll stay in the deep ocean for thousands of years or more if it ends up in the sediments. So it is a way of sequestering carbon out of the atmosphere, and it's really important for the overall climate cycle. And you can imagine just a very small change in the efficiency of that export could have really big effects on climate. So being able to predict those things is really important. So I want to focus in, I am the person that studies the carbon eaters. So I want to focus in on this respiration component of the biological carbon pump. So that previous figure was very much a simplistic view. Of course, respiration has many, many things that it goes into it. So all of this massive arrows is all different carbon transformations going between different components of that carbon cycle. And the thing I want to highlight here is that it's mainly a microbial process. So all those arrows coming out of, so the primary productivity, I have an arrow. You can't see me pointing on Zoom. Here, I'll use my mouse. All of the primary productivity is this one little box, and all the respiration is everything over here, and all of these microbes that are interacting with it and transforming organic matter into dissolved components, particulate components, some of that being able to sink out, and that's that biological carbon pump. So, you know, the size of the arrow has something to do with the amount of export that's happening. The animals and the larger organisms that we think of when we think of ocean heterotrophy are these tiny little arrows. They're a component. It's really important for, you know, the shape of food webs and the shape of ecosystems and ecosystem function. But this process is really a microbial process. And so when we go out into the ocean, we find that microbial communities have very different carbon degrading capabilities. So a big reason why we see such huge variability in the biological carbon pump is something to do with the variable functional capacities of the microbial communities that live there, and the differences that we see at different locations based off of different microbial community compositions, based off of different depths, and all sorts of physiochemical factors. So all I'm highlighting here is a couple of papers of mine that show this. So one transect that was a 7-week cruise from Uruguay to Barbados, and we saw some very particular patterns in the diversity of substrates, of carbon substrates that were able to be degraded. That, you know, some of them being persistent through depth, some of them being much more restricted to the eutrophic levels of the ocean, the sunlit parts of the ocean. And then other places in the coastal ecosystem just off of the coast of Rhode Island. This is the mid-Atlantic bight right on the coastal shelf. And we saw some really big differences in specific substrates that were hydrolyzed and their relative hydrolysis rates. So we see these very different carbon degrading capabilities depending on where you are in the ocean. And you can see this also playing out on a global scale. So this is one study where instead of looking at specific substrates or specific genes, we're actually looking at all of the genes of a very important heterotroph in the ocean called SAR86. And it turns out that you can predict the distribution of individual genes within this microbe. We had a whole pangenome of all of the genes off of from a bunch of different strains of SAR86. And you could actually predict the distribution of these genes based off their environmental data that we can get from remote sensing data. So you could predict the presence or absence of a gene based off of the remote sensing data, which means you can make these very pretty pictures of gene distributions. And what all of these rows are actually different clusters of genes that have similar geographic distributions based off of their predictability from these remote sensing things. And so you can see some very big seasonal trends in different clusters, very different geographic trends. And so we know that there are very differing functional capabilities in the ocean, and that this corresponds to some very distinct functional biogeography within heterotrophy. So these are very microscale processes. You have very specific microbes that have very specific genetic capabilities. The genetic capabilities to target these substrates tend to target very specific substrates. And there's a huge diversity of organic compounds in the environment. And so the specificity of those enzymes becomes very important. But these things accumulate over time at local and regional and then global scales to really shape the net carbon cycle and the net amount of carbon export in an environment, and really has impacts on, you know, global processes like climate. And this has shaped the Earth that we live on. The Earth, you know, didn't always look like it did today. So the biology in the oceans and in the Earth in general has really co-evolved with the Earth to shape the entire chemical environment that we live in. So from, you know, the Archean Ocean that was filled with sulfide that would be toxic to us today is now the oxygenated, beautiful, habitable atmosphere that we know and love. And now, of course, into the Anthropocene. So, there's so much interest now in how we can leverage microbial communities and biological communities to understand our environment, but also use it for, developing drugs and developing materials and all these things. And a lot of that functional capacity, most of it, almost all of it, I guess, by some definition, is in the natural environment. So when we're thinking about, doing bioprospecting to try and find compounds or functional capabilities that we want to adapt for our own uses, that is something we really need to be able to think about. So I talked about how these microscale processes are really what guide this very complex microbial heterotrophic process. But this is very hard to predict, partially because it's such a complex system, so it's very hard to predict complex systems, but also for another very important reason that I dramatically like to call microbial dark matter. So I'm sure maybe you've heard, this general thing being said before, but the vast majority of microbes on planet Earth cannot be cultured. Something like less than 1% of them have been cultured. So you go into the environment and you take that seawater and you sequence all of the DNA that's there and you try and figure out who's there, or you try and, culture a microbe, based off of certain selection material. And you see, this is a figure from a really nice paper that just went through all of the genomes that we have, including some metagenomically assembled ones, and looked at, this is at the phylum level. So each one of these rows is a phylum in the bacteria or the archaea. And the purple ones are ones for which we have N isolate, and the black ones are ones for which we have no isolates. So you can see there's a very big, there are entire phyla of the microbial tree of life that we have no cultured isolates for. And so the vast majority of microbes on Earth are uncultured. And so a lot of the ways that we tend to get at, functional inference, figuring out what things are doing in the environment, is we do sort of traditional bioinformatics approaches where we go out to the environment, we sequence the DNA, and maybe we map those reads directly to a reference database, maybe we assemble them, then map them to a reference database. But in one way or another, you get from your sequence to a comparison of all the known genes and genomes we have and try and make an inference about what that function is. And so turns out when you do this with environmental data in general, marine data is one of the worst offenders on this. Most of those genes, we have no idea what they're doing. And so that's what's being shown in this figure, and I'll just show a little bit bigger version of that. So this was a figure from the Tara Oceans expedition, which was a gift that keeps on giving to the marine science community. There was a boat that was that did a global circumnavigation of the globe and took a bunch of samples, metagenomes, metatranscriptomes at a bunch of different size classes, and have some really, really deeply sequenced samples from all of these different stations all over the world. There's also some Arctic ones that aren't shown here. And so they have very consistent sampling, very consistent processing and things. And so it's used a lot for a lot of these sort of global analyses of marine microbes. And they made a whole gene catalog. These are all the marine genes that we can find based off of these metagenomic assemblies, and then tried to functionally annotate them. And we're looking at surface, a little bit subsurface, it's called the deep chlorophyll maximum, and the mesopelagic, which is usually in the 400 to 600 meter range of depth. And the red ones are completely novel genes that have no functional annotation. So overall, it's about 30% of genes in the ocean that we have any idea what function they might do from the reference databases that we have. So in my lab, we're really focused on how we overcome that problem. And I think it's a huge opportunity for deep learning. So how can we use deep learning and some of the experimental approaches around deep learning to answer some of these big questions about how microbes are regulating ocean carbon and climate, particularly mindful of the fact that the vast majority of sequences that we have, we do not know what they do. So I think it's really useful for both modeling these complex systems and for doing reference-free predictions where we're not so reliant on these very biased reference databases that we have. So I wanted to talk about today just a few of the projects that I've been working on. There's some other applications. I did my postdoc at NASA and had the opportunity through that to think about how to apply some of these marine science concepts to space biology and astrobiology, but I'm not going to talk about any of that today. I'll talk about some of the marine science stuff. And then I'll try and figure out, I'll try and finish up with just a few sort of summary thoughts on where I think all of this is going. Since it's a keynote talk, why not? Okay, so let's jump into Looking Glass. So I think most people in the room are probably familiar with the idea of a foundation model at this point. This used to be much harder to explain to people, but now, I mean, raise your hand if you've used ChatGPT at some point this year. Like everyone, right? So it's not as hard to explain this as it used to be. But language models are kind of the closest analogy to what we do with biological sequences usually. So most of these language models, like GPT, are all trained on this masked character task, where you have, you know, a sentence and you mask out a word and you try and figure out from the context of the other words in the sentence what word could be there. And you learn to pay attention to what other things in that sentence inform that perspective and inform what word ends up going in that masked location. And you learn through that process. It's a self-supervised thing. You have basically an unlimited number of training examples when you do that, because you have just all that text and all you have to do is mask out a word. So you have lots and lots of training data, but you learn the sort of grammatical rules and the language rules that govern what goes where. And then, of course, you can learn, you can then use that in a generative way to do a very simple objective to predict the next word given a text prompt, and do that ad infinitum to generate some very, very useful things with natural language. But the key thing there is that just that one training objective and just that one generative process can actually enable lots and lots of very specific tasks like question answering or summarization or instruction following, not because you necessarily even need to fine tune your model to do that, although you can, but because it's such a good deeply learned foundation model of the rules of language that we can then do in context learning. Given a prompt, it understands what I want from it and it gives back a reasonable answer. And the reason that is because it learns these very deep features of data modality in general. And so foundation models are really what give us the ability to have big data performance on small data. You know, generally speaking, if you're training a model from scratch, a more complex system, the more complex of a function you're trying to learn, the more data you're going to need to do it. So, you know, usually the more data you have, the better your model's going to, the more data you have and the bigger your model is, the better your model's going to do. But in biology, of course, usually we're working down here in this very small data world where it kind of doesn't matter if you're training from scratch what model you're using. You don't have enough data to learn your very complex function. So, you know, you are going to do okay either way. But by getting these foundation models that are trained on lots and lots of data, and then you transfer it over to these other tasks, you can get that performance of big data on your small biological data. And that's the concept of transfer learning. So, you know, when you're training from scratch, you have your data set and you have your learning task and you have a small data. If you have a small data set, you're only going to do so well, depending on how hard your task is. But transfer learning is the idea that you can take another data set from the same data modality, so other text from language, other sequences of biology, and training it on some learning task. Any learning task will do almost. And if you have lots and lots of data, you'll probably do pretty well, and you learn that domain knowledge that underlies biology in general. And so you're able to do much better on your actual learning task that you care about with your small data set because of that pre-training of that foundation model. And so maybe the most famous biological foundation models out there right now that have a lot of buzz are these protein language models, AlphaFold or ESM2. AlphaFold came out of DeepMind and the Google folks, and both of these are designed to predict structure from sequence. AlphaFold goes through some pre-processing where they do a multiple sequence alignment. ASM goes directly from protein sequences to do that language model task, and then they find that they can predict structure from that. But there's a couple things that I want to highlight for that and why this does not work for environmental problems. And that, I mean, there's a disadvantage of something like AlphaFold where you have to do the pre-processing of the multiple sequence alignments. You have to have things in your database that look at least a little bit like your sequence. But then they're also both focused on full-length protein sequences. And in the environment, that is not at all what we ever have. We have these short sequence fragments of DNA that are, 100 to 150 base pairs long. So, maybe 1/10 of a gene. And so, and to get to anything that would look like a full-length protein sequence to use with these models, you'd have to do some assembly that introduces a lot of potential errors. And you're going to also not be able to assemble lots of things because our sequencing depth is usually not sufficient. So you'd also be throwing out a lot of data. And then, of course, my hot take of the day is that predicting structure from sequence is not really the thing that we want to do. Because structure, I mean, it has a lot to do with function, but it's not actually function. And so I think the thing that at least in the environmental context is the thing that we want to be able to predict well is function, and structure is really a proxy for that, as are a lot of other things. So enter Looking Glass, and Looking Glass is a biological foundation model for environmental sequences being these short reads that are 100 and 150 base pairs long. And so Looking Glass is trained on the kind of data that we get from the environment, these short read DNA reads. And the thing that we really want to be able to test with a biological foundation model like this, any biological foundation model, but especially this one, is whether it's capturing ecologically relevant features at all. So by my definition, that should be features that are functionally relevant, evolutionarily relevant, and environmentally relevant in the format that we retrieve them from the environment. So in these short read sequences. So this is what Looking Glass kind of looks like. So we went out and we actually did this not from raw metagenomes because we wanted to know what the DNA was for our training, but we went across the taxonomic tree of life that we have and selected DNA very carefully from, one genome from each class in the prokaryotic tree of life, and then chunked that up into these read-length DNA sequences that we actually see in the environment, and then trained it on this language model task of predicting a masked nucleotide from the context of the sequence around it. From that, it produces a embedding vector for each nucleotide, but then for the sequence overall, and then it's just trained to predict that masked nucleotide. So Looking Glass is a 3-layer LSTM, and it learns to embed through that language model task the functionally and evolutionarily and environmentally relevant features that underlie biological diversity as a whole. So before I get this question, because I always get this question, Looking Glass was an LSTM. It does do better as a transformer. We're in the process of training that again on a new and bigger data set. And my student Ratish is doing that. He's doing a great job. And as perhaps expected, it does better on every metric. So we're not ready to share that data yet, but I think the transformer is going to be a better V2. Okay, so Looking Glass, we trained it on this masked nucleotide task. And we want to see, does it capture these functionally relevant features of sequences? And indeed it does. So what we're looking at here is just the confusion matrix of the functional annotations of this pre-trained foundation model that we then just added a classification layer to see if we can predict function down to the 4th EC number. So very specific molecular function. And it has very good overall accuracy of 82%. And then this is just showing, since all you can really see is that the correct predictions in the diagonal, we show here if you take that out and then recolor it, where it tends to get most confused. based off of the true class versus the predicted class. And overall, it does pretty well, 82% accuracy. I mean, for most metagenomic functional annotation tools, going from short reads, that's pretty comparable to what we get with our traditional bioinformatics tools. And this wasn't something that was trained to be a functional classifier. It was just sort of testing whether we get those functionally relevant features out of it at all. So we are trying to turn this into an actual functional annotation tool. And my student Soumya is working on that. So stay tuned for that as well. So we capture functionally relevant features, but we also want to see if we're capturing evolutionarily relevant features of sequences. So what we did is just went out and found homologous versus non-homologous genes from the reference databases that we have, and then we split them up based off of different taxonomic levels and just said, okay, based off of the looking glass embeddings that we get out of these sequences, what's the embedding similarity? Are homologous sequences more similar than non-homologous sequences based off of just their looking glass embedding? And of course they are, or I wouldn't show you the data probably. But so here we're looking at homologous in blue versus non-homologous. And this is at the phylum level. So these are things that have very, very low sequence similarity, but they are detected in terms of embedding similarity as being homologous versus non-homologous. And we can do that with pretty good accuracy without any fine tuning at all. This is just based off of the embeddings from the pre-trained looking glass model. And that 66 to 80% is based off of the taxonomic level that we're looking at. So phylum, it does worse at 66%, but as you get more specific, it's a lot easier to tell them apart just because you do have a lot more sequence similarity as you get into more fine-grained taxonomic levels. But the thing that's, I think, really exciting about this finding is that it captures homologs that have this very low sequence similarity. So at the phylum level, 44% of sequences at the phylum level have very low sequence similarity below this sort of sequence similarity bit score that we typically use for homolog searching. That's what this horizontal dashed line is. At the phylum level, we're showing our homologous sequences and where they map in terms of sequence similarity versus their looking glass embedding similarity, and then the non-homologous sequences. And so your sequence similarity doesn't really have that much to do with your embedding similarity. I mean, there's some, well, I shouldn't say that because there is, of course, a relationship between the two. But your embedding similarity is, your embedding is capturing something beyond just the sequence similarity that we're typically relying on with our bioinformatics tools. And it's able to capture these homologs that a lot of your sequences fall into without any additional fine tuning even that your traditional tools would ignore because of their difference in the sequences. Okay, so looking glass also captures environmentally relevant features of sequences. So for this, we just looked at a bunch of metagenomes from different environments, and these are different, very broad categories of environments from the sort of metadata standards that we have for all of the metagenomes that we have out there. So we have, you know, plant-associated ones and soil and human gut microbiomes and built environment, wastewater, water, that would include your marine ones. And then just looked at how the embeddings compared to one another within those categories versus without it. And so the colors in this t-SNE plot are the different environments that we were looking at. And so you can see that they cluster fairly well. I mean, this is the self-supervised model, so these are not trained on environment at all, but you do see those differences in environment showing up in your clusters. But then of course there is a little bit of mixing between them, and this heat map shows the sort of average embedding similarity between those different metagenomes that we looked at. And so you do see places where the embedding similarities are, the embeddings are more similar than others based off of which environment you're comparing. And I think some of those comparisons sort of make sense. I mean, you have built environment and wastewater are really close together. Okay, so that's looking glass in general. But I also wanted to talk about, of course, how we can use looking glass to then apply it to the ocean and try to understand the functional potential of the ocean. And so that's where the transfer learning for microbial dark matter comes into the picture. So if you remember the Looking Glass project objectives, we wanted to do that self-supervised learning task of taking our big data set of all the DNA we have, doing that language model, masking learning task to learn those complex features that define biological diversity as a whole, but then think about how can we transfer these deeply learned features to predict functionally relevant characteristics of sequences in the environment. And all sequences, the beauty of this being that given a good model, you can take a sequence and make a prediction, and it's completely reference free. You're not reliant on your reference databases. So Looking Glass was this universal language of life. And then we take this domain knowledge that we've learned, we take our relatively small data set of our sequences with their carefully curated labels, and we see if we can get good classification out of them. So I wanted to share just a few of those transfer learning tasks we've looked into. There's, I think, infinite possibilities, and I always love hearing new people's ideas. But these are the few that we tried. So we looked into enzyme optimal temperature. Can we classify between psychrophilic and mesophilic and thermophilic enzymes? So we're looking again at just from read length sequences. Can we classify the entire enzyme's optimal temperature? We can do that with about 70% accuracy. So it's not so bad, not so bad. And we can also translate from DNA to amino acid directly because we can predict the reading frame with really high accuracy with something like 98% accuracy. So you can take any read in the environment and turn that into a little protein fragment of course, it's going to be something like 50 amino acids, but that it makes it maybe easier to interact with some of the protein databases that we have. But then I wanted to look at some actual functions in the environment. And so this is the application of just trying to predict oxidoreductases specifically. So these are EC numbers that start with 1. And if we split those up so that in the validation and test sets, you see things that are very different from what you've seen before, things that are less than 50% amino acid identity, you can predict the function of those oxidoreductases with something like 82% accuracy. So I wanted to focus on that last one a little bit for the purposes of demonstrating Looking Glass in the wild. So this is the performance of that oxidoreductase classifier model. You can get the optimal accuracy is shown with this vertical line, but of course you can get up to a precision of much higher if you're willing to sacrifice other things. And we looked at oxidoreductases specifically, partially because of my NASA postdoc that I was doing at the time, where we are really interested in early evolution of proteins, and oxidoreductases are very important for that, but also because oxidoreductases are the things that shuttle electrons around. So they're very important for metabolism, for a lot of the things related to heterotrophy. So it's a very important class of enzymes to think about energy transfer in the environment. So we took this oxidoreductase classifier and asked, how are these, if we go and apply this classifier in the environment, how are we going to see the relative distributions of oxidoreductases maybe change depending on where you are? So we took a small subsample of that TAR data set I showed earlier. So the red dots are the locations where we selected samples from different depths as well, and just applied the oxidoreductase classifier to all of the reads and said how many oxidoreductases are there relative to other sequences. And so in any of these typical samples, I'm showing one of the annotation tools that we compared it to. You have a vast majority of sequences that can't be annotated, of course. This one I'm showing with the most extreme one because it makes me look best. And of course, Looking Glass can then make a prediction for every sequence, even those that are very different from our reference sequences. And that just shows us another way. So our oxidoreductase classifier is showing its classification of, is it an oxidoreductase versus non-oxidoreductase? And then compare that to two very popular functional annotation tools, MGRAST and MyPhaser, and you see a lot of unannotated sequences. MyPhaser is a very stringent tool that looks at just those very experimentally characterized enzymes. So they're very, it's a very stringent tool. And so that's why you see, something like 98% of sequences not showing up as annotated. MGRAST is a little bit more flexible on, how close of a sequence similarity do you need to be, and its reference databases include things that have been inferred for that function by that same process, by mapping it in sequence similarities. You have those sort of error propagated reference databases in there. But you see some patterns in oxidoreductase distribution develop over latitude and depth in the ocean when you apply this oxidoreductase classifier. So here we're looking at samples from the surface versus the mesopelagic. And then this is called an oxygen minimum zone. So that's an area of the mesopelagic where you have relatively low oxygen concentrations for various reasons. And you see very distinct differences in the percent of oxidoreductases that occupied that sequence. And then you also have a very distinct latitudinal gradient as well in how many oxidoreductases relative to other sequences we have. But the really interesting thing is that when you compare this to the other functional annotation tools, those patterns are non-existent or even reversed using the traditional annotation tools compared to what we see with the oxidoreductase classifier. So that tells me that this microbial dark matter is a really important component of the microbial community. It's contributing, I mean, it's 70% of the sequences or more. It's contributing to the function of that microbiome. And maybe the way that we're interpreting our microbial communities and their function in the environment are really poorly served by some of these traditional tools that we have, and we need to be rethinking how we approach that. Okay, so well, what am I going to? I might have to skip through this. I forget. Do I have 5 minutes? Okay, well, I'll breeze through this. This is just showing how we can use looking glass in a generative way and my sort of unique take on this for trying to do this for early evolution of proteins and evolutionary trajectories of proteins. So of course you can generate from a looking glass embedding a sequence. You can see what the effect of a particular mutation has on that looking glass embedding of a sequence, and also evaluate whether that probability of having that mutation would result in a viable protein. And so without going into too many of the details of how we do that, you would expect things that are both a fitness advantage to be, things that are viable proteins, but then also enough of a functional change that you would expect it to confer some sort of evolutionary advantage. And so we see this in viral escape mutants, where they go at the top right of this plot, where we're looking at that change in embedding signature versus the probability of seeing that mutation as a viable protein. And if we look at this for the NIFD proteins, this is a very key nitrogen fixation oxidoreductase, we see some big changes in the embedding signature, that delta Z thing that I breezed past. That basically means that there is some change, functional change being conferred by, at least predicted by the model, in introducing that mutation. And some of these regions correspond to, say, the metal binding sites. So these are really important catalytic sites. So if you're thinking about, maybe looking for a new protein that does nitrogen fixation at a desired temperature or something like that, you'd be looking for a synthetic protein maybe that shows up in this top right. But you can also use this for evolutionary tree reconstruction and thinking about, okay, does it make sense if this delta Z increase conferring a fitness hypothesis is true, you'd expect that over evolutionary time, you see this increase in delta Z. And so if you do some gene reconstructions, which I won't go into, but a few random walks along this gene reconstruction, you do in fact see the delta Z of the changes in those sequences increasing over time. And so I think that's really interesting in terms of early protein evolution or in early gene evolution, where gene ancestor reconstructions really don't work very well. Could we maybe use this approach to constrain the space of possible evolutionary trajectories? And then I think there's some probably pretty obvious applications in protein design. All right, so where is this all going? In my lab, we're working on the foundation models and a lot of the transfer learning objectives, and really thinking about how to do high throughput characterization of these microbial dark matter proteins that we care about for carbon cycling. That involves some self-driving lab stuff. And then really thinking about how do we then take this back to the ocean to identify the key biomarkers or develop the machinery that we need to really do continuous monitoring of these very complex microbial processes for carbon cycling, maybe strap it to a robot and see if we can get continuous data. So I think there's lots and lots of applications out there. And we're working on all these. Would love to talk to people about more of them, but I will wrap up because I'm running out of time. And yeah, happy to talk more.
Oh, that was great. I enjoyed it. So I was wondering about your looking glass based embeddings and how it's kind of capturing sort of the space, the geographic location, right, of these. And then sort of also similar to what Arvind was talking about with this GenSlim, but the SARS-CoV-2 embeddings were, capturing somehow the geographic location. So what are your thoughts on how to really go, retrieve what information is contributing to that classification? It sort of gets to the explainable AI aspect of it, which we're not covering that much in the symposium, but what are your thoughts on how to really explain those?
Yeah, I mean, there's lots of explainable AI approaches out there. I didn't show any of our projects around causal learning today, but I've been involved in some projects trying to think about, how do we select those features that are not just driving the predictions, but also like causally associated and not just correlated with that outcome. So I think that's one approach. And there's a whole sort of field of causal machine learning around that. I mean, there's lots of other sort of feature identifications approaches around explainable AI. I think some really, we're doing a little bit of early work on how to use language models to try and explain some of our predictions and, using multimodal models that include a language model component to both make better predictions, but then also explain those predictions. So I think there's lots of possibilities.
Thanks so much. That was great. In terms of the input to your looking glass, I'm curious, you talk about short read Illumina sequences. Is that an exact like 100 base pairs, use some kind of K-mer? And I'm just curious when you expand and contract that, if you notice, because clearly the context around that is critical. And also I'm kind of nesting another question there. What do you think the repercussions of longer read, high fidelity sequencing are for this type of model?
Yeah, so the sequences that went into the actual training of the model were, so we created a distribution of like realistic sequences. So they were between 60 to 150 base pairs with an average read length of like 120 or 130, I think. And yeah, so I think they were trained on realistic sequences, and you do have a context window size that we tuned, and I think it was at 128 ultimately. So something, because you don't want to go above, once you go above your sequence length, it doesn't make any sense to use a context window above that. So in terms of the implications, I mean, I'm very interested in the question in the case of looking glass, how big of a sequence can you go in? Some of the data I show you actually do, like the homology was done on full length gene sequences, because otherwise you would get things from totally different parts of the genes, and it is decently high fidelity. But some of the like protein language models or gene length models or genome length models, where do you maybe see worse performance as you lower that sequence length? I mean, there's only so much that a read can tell you. It's the environmental sequence that we have. And so I think it's very important that we look at them, but there is some limit of information that it can capture. So I think there's a trade-off there, of course, and I'm very interested in like where does looking glass break down in terms of the length of sequences as you get into bigger sequences versus some of these other models that are trained on longer sequences, and where they break down, and is there, what use cases are best for which model is better for which use cases, I think is very dependent on your question. But I don't know if that really answers your question, but yeah.
Hey, that was a great talk. So I think you may have mentioned this, but I missed it. But what are some of the features of protein sequences that give us more insight into their function as opposed to protein structure?
I mean, that gets back to some of the explainable AI stuff. I don't have a really good answer for that right now. I didn't show it today, but we did do like a sort of feature analysis, highlighting which nucleotides are driving the prediction and which ones are, which nucleotides are most strongly selected on. You see that a little bit with your delta Z map, where you get some mutations that have a big effect on the function. You can use that to try and get at it. But it's a little bit, at this point, a little bit of a manual interpretation process. Like, oh, well, those big delta Z changes correspond with catalytic sites. So that kind of makes sense. But yeah, I think it's a multitude of features. I think it has to do with the evolution of those enzymes. And I think it has to do with structure. And I think it has to do with, other features, GC content, things like that. Yeah.
Really interesting talk. I have a couple questions about these DNA models. I can wrap my head around protein models. The DNA, there's like frame shifts, and you have data genomics data, and it's like short fragments. I guess one question is, how do you tokenize these? Do you just work with the raw base pairs? And then also, when you construct your training, do you also truncate it to be frame shifted or complemented or anything like that?
Yes. So it's a single nucleotide model. So it's a very small vocabulary of the 4 nucleotides and like a end of sequence token. And I tuned different Kamer lengths and different, step sizes if you wanted to have them overlapping or not overlapping. Ultimately, I thought single nucleotide it performed best, and I think it made the most sense in terms of not introducing too much redundant information. So you could train on less data, but what was the other part of your question around how it, oh, in training, yes, yeah. So this was, so we cut it up into different read length sizes, and then, yeah, it was selected randomly based off of where it started. So you have all different reading frames. It was a flip a coin, whether it was reverse complement or not. So yeah, did our best to make it look like real reads.
Hi, great talk. I was just wondering, because now you're kind of really getting into the microbial genomes that can be cultured, and we don't know a lot. So with the oxidative reductase being such an important class of molecule, are you finding with your two like new classes, or are they kind of all something that have been annotated? Like, are you finding new divergent ones, and things like that? Like, because I'd assume you're really expanding on the sequence diversity from this work, right? Or I might have just gotten it wrong.
Yeah, that's a really good question. That's still ongoing work. That's a lot of what Simi's doing. She's creating that new functional classifier and then looking at applying that to our metagenomes as well as that gene catalog that I mentioned, and just seeing what those classifications are and what those embeddings are and doing that analysis around how novel are they. But I can tell you that marine gene catalog, there are things that have very little similarity to things that we have. And I don't know if anyone's done the gene trees associated with that, partially because there isn't a particular function associated with them, so it's hard to do, but I'd be willing to bet that yes, but I don't have a good answer for that right now.
So is this something that's like available at the database that we can like access as like people who look at protein sequences, especially?
The gene catalog?
Yeah.
Yeah, it's called the OMRGC, the Ocean Microbiome Reference Gene Catalog. And I think their website's like oceanmicrobiome.org or something like that. And so it's that Sunagawa et al. If I can find it, paper. And that's that gene catalog that they made. And yeah, it's available online. You can download it.
Thank you.
Yeah.
A very interesting topic, and I have one high-level question is about like your model, the looking glass. You use it to predict when in the training phase, it's trying to predict the nucleotide sequence, right? And always people find surprise. It also have the information of the embedding similarity, which along with the clone tree. So my question is, do you find this is a common thing, like you use something to training for different purpose, and finally you find the embedding is very useful in another task? Is something, have you tried different pre-processing step and finally find one embedding way that specifically work, or it's very common thing you train on one task and in the second task it has a good performance?
I mean, I think the general phenomenon of being able to take a foundation model and transfer it to a new thing, that's very common. And I think we've seen that in biology a bunch now. I think there are lots of examples of that. Yeah. So I think so. I don't think that, I think the jury's out a little bit on, what's the best way to encode biological information, especially in reads. I don't know anybody else who's working with reads. So, do you want to do anything different than just your transformer? I think because they're actually relatively small, you don't need to worry about the larger scale patterns where maybe you want to add a convolution to it or anything like that. Like, I actually think the transformer makes a lot of sense for short reads, but yeah, I think there's a lot of questions out there, and a big interest of my lab is thinking about how to go beyond the sequence level where we're taking a sequence and we're making a prediction to a whole microbiome level. And I think that introduces whole other architectures that are very open questions of how to do that. I think graphs has something to do with it, but.
Thank you.
Yeah.
Any more questions? If not, let's thank Dr. Hoarfrost. Thank you.
Thank you so much.
Before we conclude the section, I just want to remind you that right after this, we have our lunch from noon to